हेलो 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 गाय हेलो हेलो आवाज तो जाता उमन नहीं आवता लेकिन हेलो हेलो हेलो
हेलो हेलो हेलो हेलो हेलो आवाज आवत क्लियर आवत एकदम ना कोनो दिक्कत नहीं बा नहीं अब तू बोले हां अब तू बोल हेलो
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 आवाज जाता बोलो
हेलो 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 ठीक
बोलिए सर।
Hello, good morning everyone. Voice is clear? Okay, good.
ये तो इसमें नहीं आ रहा था ना, इसमें वो ऑडियो नहीं आ रहा था ना।
हाँ,
बस कर दे रहे हैं, अभी बैठे रहिए सर।
चेंज करेंगे इसको।
नहीं चेंज नहीं करेंगे, बस इसको चेक करेंगे। आप बोलिए सर।
हेलो। हाँ, गुड मॉर्निंग सर। हेलो। हाँ,
आवाज क्लियर है ना एकदम?
हाँ।
कोई
गुड
खिड़खिड़ा नहीं,
मॉर्निंग।
कोई दिक्कत नहीं है ना सर?
नहीं नहीं, ठीक है।
ठीक है।
कर लेंगे। सही कहूँगा। सर बोलिए। हेलो, हेलो। आ रही है। तुम बाद में आवाज आ रही है। सबसे बात हुआ था। अब क्या हुआ? सर को फोन कर दीजिए। बारिश सर, अभी ना आपको एक मेल किया कोई जरा। बारिश सर को है ना सर, रिकॉर्डेड वीडियो पर जल्दी भेज दीजिए। हाँ, वो बोले यदि मैं नहीं होता हूँ, एक लड़के इसको बोला क्यों नहीं उठाएंगे, बताइए। फिर भेज देंगे। क्या चाहिए बताइए। क्या वीडियो देखिए, सुनिए। शेयर मत कीजिएगा। ठीक ठीक से समझ लीजिए, एक ही है। तो अपना नहीं खोलो वहाँ पे, उनका एक्सेस प्रोवाइड किया होगा, फिर डाउनलोड हो सकते हैं। रिक्वेस्ट एक्सेस कीजिए ना। तो तुमको तो एक्सेस दिया होगा, तो अपना ये ईमेल ईमेल डाउनलोड नहीं, डाउनलोड तो करने पड़ेगा। क्यों डालना है?
हेलो। हेलो। ये तो ठीक है।
हेलो चेक चेक ये मत लगाइए नहीं तो चेक चेक हाँ हटा दीजिए ये सर हेडफोन लगाइए सर
विपिन हेडफोन लगा देखो आवाज आ रहा है बोलिए
हेलो
हेलो हेलो एम आई ऑडिबल
क्लियर है ना आप
हेलो यस हेलो
पहले वाला अच्छा था ये वाला अच्छा नहीं
हेलो कौन बात कर रहे हैं
ठीक चार्ज वाला है क्या चार्ज करके रखो
ये वही है
हाँ
पहले वाला ही
हाँ
इसको चार्ज लगा दे रहे हैं
ठीक है ताकि यूज हो जाए
ताकि तुरंत हम
ठीक है कर दीजिए कंप्यूटर में लगा दीजिए उससे चार्ज हो जाएगा
उसी मतलब
एडाप्टर
दूसरे में लगा देंगे
हाँ कंप्यूटर में लगा दीजिए तो उससे चार्ज हो
हाँ।
जाएगा यूएसबी
अरे और
भोजपुरी सुनना
थोड़ी ऐसा बदनाम कर दिया नहीं कहाँ ये
ये वाला जी 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 जरा शेयर कर दीजिए वाला स्लाइड। सर 10 बन गया क्या?
यस,
सर स्टार्ट कर सकते हैं सर।
3 minutes रिमेनिंग। हाँ, किसका है? ओके 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 ओके।
वो उससे वाला दिक्कत आ रहा है उसमें कोशिश की।
वो कर देंगे।
और अभी ये अगर इसको ये करते हैं तो बंद बंद करके जितने सब पार्टिसिपेंट सबको दिखना है।
हाँ ये स्लाइड्स स्टार्ट कर देते हैं। अभी पूछिए
कितने लोग ज्वाइन हुए? Good morning, all of you. I welcome again on the third day of this workshop. Today, what we are going to do is start with the practical session from Dr. Paresh Kulkarni, and then followed by different sessions from Dr. Gushet sir, and then a session on, again, one practical session on thromboelastography by Mohammad Iqla. So these are the programs scheduled for today. First, we'll be starting with the intravital microscopy. It is one of the very important techniques in the thrombus biology that you have seen in the lectures from Dr. Manas Kumar Nayak yesterday as well. These are very good techniques because these will show actual thrombus formation and also the effect of different type of anticoagulants and antiplatelet agents. So these are very good for publication point of view, and also they will show you your effects of different type of therapies on the real time mode. So these are generally done on murine model, and this will actually emphasize the different type of anticoagulant therapies which are available, and also we are able to screen for the candidates for new drug therapies and other type of herbal or other medication, whether they are effective or not. So, and being a good documentation technique. So we'll be starting with video from Dr. Paresh Kulkarni. So let me introduce Dr. Paresh. He is one of the colleague of mine. He joined this Institute of Medical Sciences, Department of Biochemistry in 2016. He has completed his MD and PhD, both from biochemistry, from Institute of Medical Science under mentorship of Professor D. Dasser. And he is a very eminent worker and also has got many accolades and awards. He has uh, almost 15 publications in various important reputed international national journals. He is a member of National Academy of Medical Science (M.A.M.S.), and also he has been selected for Research Award of I.M.S. 2022. So let me start with the video from Dr. Paresh.
A very good morning to all the participants. My name is Paresh Kulkarni and I am an associate professor in the Department of Biochemistry here at Institute of Medical Sciences, B.H.U. Today I shall be speaking about mouse models for intravital imaging of thrombus formation. That is, I'll be speaking about ways in which you can see formation of thrombosis in intact vessels using a fluorescence microscope. So here the model which we are using, the mouse model, the mice is alive, the vessel is intact, and then you try to image thrombus as it forms inside the vessel using a fluorescence microscope. So that is what I will be speaking about today. So before I begin to describe about these mouse models for intravital imaging of thrombus formation, let us know what an animal model is. So an animal model is a living organism which can be used to study fundamental rules of biology or behavior, or spontaneous or induced pathological processes, and in which phenomenon in one or more respects resembles the same phenomenon in humans. So you can either be studying the physiology, or you can be studying diseases, or you can be looking at processes which are similar in many ways to what is happening in the humans. Okay, so one of the models, disease models for thrombosis, an ideal model would be should be as similar to the events happening inside a human being during thrombus formation as they could be. Okay, so what are the characteristics of an ideal mouse model of thrombosis? Number one, it should simulate the causative events leading to some form of clinically relevant thrombosis. In human beings, thrombosis or clot formation within the blood vessel lumen can happen usually under two circumstances. One, where there is an atherosclerotic plaque which ruptures, leading to formation of thrombosis at the site of plaque rupture. Second, there could be stenosis or narrowing or stasis or lack of blood flow in the deep veins in the lower limbs mostly, leading to a thrombus formation. So these are the two clinically relevant ways in which thrombus can form. So your model should have an inducer which is very similar to a plaque rupture or a stenosis or stasis of blood in the deep veins. Second, it should be done in a vessel anatomically comparable to one wherein clinical thrombosis occurs. So we all know where arterial thrombosis mostly occurs in humans. It is either in the coronary arteries leading to what is known as acute myocardial infarction or heart attack, in the carotid arteries or downstream middle cerebral arteries or other vessels in the circulation of the brain, or vessels in the lower limbs, especially the deep veins, where there can be formation of a thrombus. So the vessel that you choose to study thrombosis in animal models should resemble the characteristics of the coronary artery or carotid artery or middle cerebral artery and so on. Third, an ideal mouse model should generate relevant and consistent quantitative data. So there should not be a lot of subjectivity in interpretation of the results of your experiment, and the result should be reproducible as well. So and the fourth most important characteristic of the ideal mouse model of thrombosis, it should provide as much information for the dynamics of thrombosis as possible. Okay, thrombosis, there are a lot of dynamics in thrombosis as relates to extent of platelet activation, growth of thrombus, thrombin generation, fibrin clot formation, clot retraction, embolization, So all these dynamics should be captured by your animal model of thrombosis. So what are some of the important mouse models of thrombosis? These can be divided into two classes: the induced or experimental models, or the spontaneous models, usually in genetically modified animals. So normally mice do not spontaneously develop thrombosis. They develop thrombosis only when they express certain human proteins which are important in thrombosis, such as plasminogen activator inhibitor 1 or Pi1. So the only mouse model for spontaneous thrombosis is transgenic mice expressing human Pi1. The induced or experimental models are more commonly used because we can control the extent of injury, as well as we can decide the site of injury, and we are able to image or quantify the extent of thrombosis which is happening. So induced or experimental models can be triggered by various stimuli. So these stimuli can be chemical, biological, physical, or surgical. Example of chemically induced thrombosis models include ferric chloride injury induced model of small or large vessel arterial thrombosis, rose Bengal dye photochemical injury induced model of large vessel arterial thrombosis. Example of biologically induced models of thrombosis include collagen epinephrine induced model of venous thrombosis and pulmonary embolism. Physically induced models of thrombosis include laser injury induced models for thrombosis as well as hemostasis, and ultrasound induced atherosclerotic plaque rupture and thrombosis in ApoE deficient mice. The fourth type of induced models is surgically induced, and the example is IVC ligation induced model of deep vein thrombosis. So IVC refers to inferior vena cava. So each of these models, the trigger for thrombosis is different. So let us now understand what is the mechanism of thrombus initiation in some of these models of thrombosis. In rose Bengal or photochemical injury model, you inject a dye known as rose Bengal dye into the veins of mice, and you select the area where you want to visualize or measure thrombosis. So usually this is the carotid artery, and then you expose the site at which you want to induce vessel injury to a green laser light. So this green laser light leads to excitation of rose Bengal dye and formation of singlet oxygen, which causes oxidative injury to the vessel wall. The second type of experimental model, ferric chloride injury induced model, involves severe free radical
I request all
injury induced
to please mute
endothelial damage and denudation. So you can see here both ferric chloride injury and rose bengal photochemical injury involve injury to the vessel wall through oxidative stress. However, rose bengal photochemical injury is a milder form of vessel wall injury compared to ferric chloride. In collagen epinephrine induced model, collagen and epinephrine, which are known stimulators of platelet activation, are injected into the veins of mice. So there is activation of platelets and thrombus formation in the veins of mice, and these thrombi embolize to lung vessels or pulmonary vessels, leading to pulmonary embolism. Laser injury model involves exposure of small vessels usually to laser light, which leads to damage to endothelium due to heating. So this injury is fairly limited and there is little morphological change to the vessel wall. So there is some endothelial injury, but there is no endothelial denudation. In ultrasound induced atherosclerotic plaque rupture, you put a ultrasound probe near a atherosclerotic plaque in the carotid vessel, and this ultrasound probe leads to rupture of the plaque and release of the contents of the plaque, as well as exposure of the subendothelium, leading to simultaneous activation of both platelets and coagulation, which is very similar to what happens in humans during heart attack or stroke. IVC ligation induced model, the mechanism of thrombus initiation is stasis, that is lack of blood flow. So let us go. So the two most commonly employed models for arterial thrombosis especially is ferric chloride injury induced model and laser injury induced model, which are also employed for intravital microscopy of thrombosis. So let us dig deeper into the mechanism of thrombosis in these two types of mouse models of thrombosis. In ferric chloride injury model, when you apply ferric chloride to the mesenteric arteriole, the ferric chloride seeps into the vessel wall and then reaches the endothelium. It causes oxidative damage to the endothelium, leading to exposure of the subendothelial collagen, which is, and also von Willebrand factor, which is bound to this collagen. So this leads to platelet adhesion and rolling on von Willebrand factor initially. So as they firmly adhere to the collagen, they then secrete ADP and thromboxane A2, which leads to recruitment of more platelets into this thrombi. These recruited platelets or activated platelets then expose a protein known as P-selectin on their membrane. P-selectin in turn interacts with PSGL1 or P-selectin glycoprotein ligand 1 on monocytes, neutrophils, and extracellular vesicles derived from monocytes and neutrophils, which carry a protein known as tissue factor. Tissue factor exposure leads to activation of extrinsic pathway of coagulation, which ultimately leads to generation of thrombin. Thrombin cleaves the fibrinogen in the bloodstream to form fibrin clot. So there is formation of both a platelet thrombus as well as fibrin mesh, which holds this platelet thrombus together. So this was about the mechanism of thrombosis in ferric chloride engineering model. So how do we do this intravital imaging of ferric chloride induced thrombosis? What we do is we anesthetize the mice and then inject fluorescently tagged antibody against platelets into the mouse veins, which labels a significant proportion of circulating platelets fluorescently. Then we do an midline abdominal incision, exteriorize the mesentery, and then we isolate one of the mesenteric vessels which supplies blood to the intestine. So these small arterioles are usually of the size of 100 to 200 microns. So these mesenteric vessels, there is both a vein and an artery associated with each other. So the artery is smaller in size, but it has a thicker wall. So you need to focus this artery under the microscope, which is attached to the camera, so that you can capture the images on a computer. When you illuminate the blood vessel with a intense light and then apply ferric chloride to that site of focused mesenteric arteriole, so there is denudation of the endothelium and platelet starts adhering and forming a thrombus at the site of injury, and finally they form a large clot which occludes the vessel. So all this process can be recorded with the help of a camera attached to the microscope, and with this video of thrombus formation can be analyzed later for assessing different characteristics of thrombus formation. So this is an example of ferric chloride induced model of
मेरे को
arterial
क्या 10 दिन
thrombosis.
4 दिन
This
का
is a
है वो
video
सोमवार
of the
से
thrombus
मंगलवार से
formation.
शुक्रवार
On the left side you
कल और
see
रहेगा
control mice and on the right side drug treated mice. So you can see two vessels here,
नहीं नहीं
a
नहीं
larger vein and a smaller lumen artery on the right side. So you can see here platelet thrombi forming and growing in the control mice, while there is no or little adhesion of platelets in the drug treated mice. So you can, whatever green labeled things you're seeing in the control mice are platelets which have been fluorescently labeled. So this is how a ferric chloride induced model of arterial thrombosis looks like under a fluorescent microscope. our injury model is slightly different. Move on to the second model of arterial thrombosis, which is the laser injury model. So the mechanism of thrombosis in laser injury model is slightly different. Here there is no denudation of endothelium. The laser leads to heating up of the endothelial layer, which causes some endothelial activation and injury, but the subendothelial matrix is not exposed. So this laser injury leads to expression of tissue factor and thrombin generation. Thrombin generation activates the platelets. The platelets then form a thrombus at the site of endothelial injury. So here the initial trigger is the thrombin, while in ferric chloride injury the initial trigger was collagen and von Willebrand factor bound to collagen. So while ferric chloride injury and thrombosis happens over several minutes, sometimes up to 30 to 40 minutes before occlusion. In the laser injury models, the dynamics is faster. You see a clot forming within 1 or 2 minutes and then it gets embolized. So here the dynamics of clot formation is much faster than in the ferric chloride injury model. So this is how a laser injury model looks like. So apart from the microscope, so the objective is also attached to a laser board with different laser sources. So these lasers can be used for both imaging as well as inducing the injury. So you induce injury with laser and then record thrombus formation using a camera attached to the microscope. So this is how a laser injury model looks like. On the left side is the control and the right side drug. So you can see here clot forming and disappearing in drug treated mice, while there is a more stable thrombus in the control mice. Moving on to the third model, which is the ultrasound induced atherosclerotic plaque rupture and thrombosis. Here the mechanism of thrombosis is different from the ferric chloride induced model in only one way, that the subendothelial matrix which is exposed in this model, the structure of the plaque collagen is altered, while the structure of collagen exposed after ferric chloride injury is normal. So this altered structure of collagen in the atherosclerotic plaque leads to different signaling cascade compared to the signaling initiated by collagen exposed after ferric chloride injury. So you can look at signaling pathways which are specific to that initiated upon exposure of platelets to plaque contents. So one such important molecule is Button's tyrosine kinase, and inhibiting this enzyme leads to selective inhibition of thrombus formation induced by atherosclerotic plaque contents. So here you can this type of treatment can lead to prevention of thrombosis at the site of plaque rupture, while sparing hemostasis which occurs at other sites of endothelial injury in the body. So the bleeding risk is reduced while thrombosis is prevented. So ultrasound induced atherosclerotic plaque rupture and thrombosis model usually is done in carotid arteries and in genetically modified mice which lack a apolipoprotein E. So due to the lack of apolipoprotein E, so there is accumulation of fat in the blood vessel wall, leading to atherosclerosis and atherosclerotic plaque formation. So this atherosclerotic plaque in the carotid artery is ruptured by using an ultrasound probe, and then using an fluorescent microscope you image thrombus formation at the site of plaque rupture. So this is how an atherosclerotic plaque rupture induced thrombosis looks like. So this is in carotid artery at the site of bifurcation. So what are the different parameters which you look at while assessing your video images of platelet thrombus formation in these different mouse models of thrombosis? In some of these models, especially ferric chloride induced mesenteric thrombosis as well as laser injury induced mesenteric thrombosis, you will be able to look at single platelet vessel wall interactions, which is determined as the number of fluorescent platelets that are deposited on the injured vessel wall during 1 minute, beginning 2 to 3 minutes after injury. So you simply count how many platelets adhered to the injured vessel wall during a period of 1 minute. So this period of 1 minute is chosen usually 3 minutes after the injury. So when you have already removed the ferric chloride soaked paper which you have used to induce injury. Then you also look at something known as time required for first thrombus formation. So usually the size of the first thrombus is arbitrarily defined as 30 to 50 micron, depending upon the size of the vessel. So it could be 30 micron for mesenteric arteriole or 50 micron for carotid artery. So depending upon vessel, you can arbitrarily decide what is the size of first thrombus that you want to assess. Then you look at what is known as thrombus growth rate, which is the rate at which a thrombus of size, say arbitrarily defined size of 30 to 50 micron, grows further in size over the next 3 minutes. So you look at thrombus diameter at 0 time when the thrombus size was between 30 to 50 micron, and then you look at the diameter 1 minute later, 2 minutes later, and 3 minutes later, and then you see what is the change in diameter per unit time. So this is known as thrombus growth rate. Then you also look at thrombus stability, which is determined by measuring the number of thrombi of diameter larger than 30 to 50 micron embolizing before vessel occlusion. So when the platelet thrombus formation occurs at the site of injury, sometimes the thrombus is not stable and platelet aggregates dissociate from this thrombus mass and embolize further. So you can count the number of times these masses of platelet aggregates dissociated from the main thrombus and embolized before there is complete occlusion of the vessel with thrombus. So this gives you an idea as to how stable the thrombus is. There are certain drugs or certain enzymes or proteins which affect thrombus stability and not thrombus growth rate or time to occlusion. So in these special circumstances, you might want to look at thrombus stability. The fifth and most commonly employed parameter for assessment of thrombus formation in mouse models of thrombosis is time to occlusion, which is the time required for blood to stop flow completely for 30 seconds. So this is time to occlusion. So on an average, in ferric chloride induced mesenteric thrombosis, this is around 20 minutes. It could be delayed further in rose bengal or photochemical injury induced model, where it could be somewhere between 30 to 40 minutes, and it could be even lesser in cremasteric vessel in laser injury model, where it is in few minutes. You can also look at the site of vessel occlusion. So whether the occlusion occurred at the site of injury or downstream. So if it is occurring downstream, it means that the embolization has been more frequent. So the thrombus stability was affected, which lead to thrombus occlusion downstream of the site of injury. So this was all these parameters can be assessed by fluorescently labeling platelets. However, intravital microscopy can not only help you look at platelet thrombus formation, it can also allow you to do evaluation of thrombus dynamics by looking at different characteristics of a thrombus. You can look at extent of platelet activation in different regions of thrombus by fluorescently labeling platelets with p-selectin. So you can dual label platelets with a platelet specific marker and then a platelet activation specific marker against the p-selectin. You can look at extent of fibrin formation by fluorescently labeling fibrinogen or fibrin. So you can also look at thrombin generation and the localization of thrombin activity within the growing thrombus using a special probe. So this is usually a peptide which is a thrombin substrate, which is linked to a antibody against platelets. So it will localize to the site of thrombus. And then when the thrombin cleaves this substrate, that is peptide, the fluorescent tag attached to the peptide is released from an adjacent quencher. So as the quencher moves away from the fluorogenic peptide, so there is increased fluorescence, which can be recorded as thrombin activity in different regions of the thrombus. We can also look at platelet packing density and solute diffusion rates by fluorescently labeling albumin with caged fluorescein or by fluorescently labeled dextran. So these are the different ways in which you can look at thrombus dynamics by intravital imaging of thrombosis. So in a subsequent session, you will see a practical demonstration of intravital microscopy of ferric chloride induced mesenteric thrombosis, and you can understand better as to how intravital microscopy of thrombosis works. Thank you.
Hello. So you must have watched the video from Dr. Paresh Kulkarni, who has explained in his lecture that how intravital microscopy is being utilized for different type of coagulation studies. Now, if you have any question, please feel free to ask, and our colleagues will be able to answer them all. If I Hello, please ask your questions now. Please unmute everybody.
Very good morning, sir. I am Shritanka Prasad from Manipal Tata Medical College, sir. Sir,
Yes.
the term is intravital microscopy. So, what is the meaning of this intravital, sir? Is it because we are observing the fluorescent level thrombus in the form of the platelets? Means why that term is, sir, intravital?
Dr. Prasad, to respond to your query,
Yes, sir. Thank you, sir.
yeah, intravital means that while the animal is alive, okay?
Yes, sir.
So throughout this procedure, the animal remains alive, and all the hemostasis process is on. Physiological hemostatic process is on while thrombosis is being recorded. So any such procedure which is done while the animal is alive is known as intravital procedures. So in this case, intravital microscopy. Is it clear now?
Thank you so much, sir. Yes, sir. Yes, sir.
Most welcome. Although whoever will be asking, just first introduce yourself, and if possible, just keep your video on. Perhaps we can look at you also, see at you. That will be better.
Very good morning, sir.
Good morning, sir.
Am I audible?
Yes, you are.
Yeah, thank you so much for a very informative talk. And I have a few questions regarding this ferric chloride treatment with animals. So did you found any abnormalities with auto treatment with ferrichloride other than this hematological parameters like, organs and all?
Sir, we are not injecting the ferriculide or not treating the ferriculide to the direct mice. We just
Okay.
take a 10% of ferriculide solution and dip with the tissue paper, and that tissue paper we will place on the mesenteric arteries directly. And after 3 minutes, we just remove that tissues. So no need to inject and no need to treat for the incubation.
Oh, I mean, since it is a chemical in nature, but definitely it has a side effects and all. No? So that's what I'm thinking. And one more question is, did you found any changes in drastic changes in ROS and reactive oxygen species because of this ferrichloride treatment, especially in platelets and all?
Sir, for the ROS, sir, we have to check, collect the blood sample, and in intravital microscopy, we are not because we are not inducing the ferrichloride at all the level in whole the body. We specifically target at a particular sites. So we just focus on the injury site and formation of thrombi and activation.
First of all, let me explain that for that we expose first the mesenteric artery. So lay it open under the microscope. Then whatever the region that we use, that we shall induce the thrombus. We shall place a drop of just by touch a drop of ferrichloride there locally. Allow some time, and that thrombus will be, in the process thrombus will be formed because platelets are already made fluorescent before the incision takes place. While the mouse was there without incision, already by tail vein we have already injected fluorescent level antibody, and platelets have circulating mouse has circulating platelets which are already fluorescent. Now then we make the decision, open the mesenteric artery, lay down a drop of ferrichloride there, let the thrombus happen, and as the thrombus will happen, the platelets will make thrombi, and because they are fluorescent, it will be recorded in the under the microscope. And this is a live recording while the animal is alive and thrombus is getting formed. We can study the effect of drugs in another animal in parallel.
This will become more. This will become more elaborate when you will be watching the next session, which is the live demo for this particular therapy. Okay, so particular experiment. So it will be more evident to you, and then ferrichloride will be going through the artery by simply by diffusion. So it's a very local process. Okay, so it will be more elaborate to you when you'll be witnessing the live session. Just it will be happen next.
Tell the name of the person was asked.
And one more, the last question, sir. And whenever you were talking about thrombus formation and all, but the generation of the platelet will be starting from the megakaryocyte. That is already known that. So did you found any abnormalities in megakaryogenesis? I mean, either hyperproliferative, I mean, reduction in the number of kind of thing. And moreover, what about this stress in that condition? So, I mean, is it possible to working with this animal model? This kind of, I mean, the pre-stage of platelet formation.
I'm not able to get your question. Please repeat your question.
See, this thrombus formation is, as you were saying, that is basically accumulation of platelets and all, right?
Yes.
So the platelets are basically from arise from the megakaryocyte, from the bone marrow in it's a process. So what is about your opinion on this megakaryocyte, whether they will get affected by this treatment or anything like that?
No, sir. It is only because we are using a ferrichloride only locally on the mesenteric arteriole. So local endothelium will be denuded, and that will elicit thrombus formation locally. So the animal is otherwise not affected. This ferrichloride is not going into the system, right? So if you inject ferrichloride to the system, in no time the animal will die, because clog the system. It is a poison for the animal. So here is the exterior externalized artery, we just put a drop. So we are not going to study effect of, we are no way to study that megakaryocyte, how it liberates platelets. That is not being studied here. Is it clear?
Okay. Okay.
Uh,
Thank you so much. Thanks
there,
for
there,
explanation. Yes.
yeah, there is one question in the chat box that how we are labeling the platelets.
Sir, one more question from Vishnu Priya.
Yeah.
Which fluorescence agent is used to color platelets in intravital microscopy? And last is from Pooja Yadav, and similar question here. So for the intravital microscopy, we are using two different strategy. First, in my lab, at the time we are using fluorescent tagged platelet specific antibody which labeled which named Alexa Fluor X488, which is purchased from the Emfred. And this antibody specifically labeled the platelet glycoproteins, by which we can detect the platelets activity in intravital imaging. And if we do not have this antibody, so we can also track the movement of circulation of platelet by staining by the calcine AM. If we want to stain with the calcine, so first we have to collect the blood from the mice and then isolate platelet, and in that platelet we add the calcine AM, and this calcine AM stained platelet we next injected intravenous into the mice, and after the injection then we can also track the uh, thrombus formation in mice.
Good morning, sir. Sir, I have one question.
Yes, ma'am.
Introduce yourself.
Please introduce yourself.
Myself, Vishnu Priya from IIT B.H.U.
Yeah.
Okay.
Sir, can we use the ferrichloride model in the carotid artery?
Yes,
You are talking about murine model, right?
Yes, sir. Like
In the
you have used in mesentery arteriole. Can we use this model in carotid artery?
I'll just intervene. The mesenteric artery is blood flexible one, and it is able to place that artery on the microscopic point of view. Carotid artery will be a bit tricky because it will not be able to place it on the field of view. Okay, so that will be a bit tricky.
It can be done by using a upright microscope.
Yeah. In our laboratory setup, there is an inverted microscope. So, but it can be done with upright microscope.
People are doing that thing also.
Yeah.
So
Okay,
you
sir,
can
sir,
use.
one more question.
Yeah.
Like this ferrichloride method, somewhere I learned that these method only is sensitive for anticoagulants and antiplatelets. So can we use the fibrolytic drugs also for for their effect, to measure their effect in this model, ferrichloride model?
So as you listen to the talk by Paresh, so the whatever color you are seeing, they are platelets, because platelets are leveled, right? But because locally thrombin is also released by extrinsic system, thrombin also induce fibrinolysis and fibrinogen conversion to fibrin, clot formation locally. So if you give anti-fibrinolytic drugs to study their effect, then there may be some embolization instability in the in your view of the clot that is formed. Stability may be compromised. But it is the best system to study antiplatelet drugs, because what you are looking at are directly at the platelets.
So is there any model for fibrolytic drugs, sir, for thrombotic?
Fibrolytic, so you see, fibrolytic, you can study many intro systems. We are
for the fibrinolytic assay system, we are in my lab we are using the thromboelastography. So today in today's session we will discuss about the thromboelastography. So in that video you can get your answer.
Sir, one last question. can we why we are not using this model, FeCl3 model, to the veins?
Veins?
Because
Only for arteries, why it is not for veins?
our theme is to see the thrombus and yes sir.
You see that the thrombosis can be found within the veins as well as within the arteries. But arterial thrombosis is platelet rich. But the thrombi that is found in the veins, they are more rich in protein part, that is fibrin rich thrombus. So here, because we are studying the effect of any drugs on the platelets, so it is the fitness of things that they are studied in the arterioles, and it is easier
Yes.
also, I think.
Yes sir,
based
it
on-
is easier.
it is more practicable to do it in arterioles.
Okay, sir. Thank you.
There are deep vein thrombosis models also. I think yesterday somebody spoke that. Yeah.
Dr. Manas sir.
Manos.
Sir, I'm from Mananmanim Sundanar University, Tinelveli. Like, I just want to know that whether this intravital microscopy is used in clinicals, that is on human, or only it is used in laboratory purpose.
No ma'am.
Ma'am, as we have learned with the ethical point of view, ma'am, we will not be doing any harm to any human subjects. Okay. So, and as you know, arterial thrombus can be life-taking. Okay. And many of the animals that will be using here, ultimately we need to euthanize them. Okay. So, this will not be an ideal model for humans.
It is never a model of for humans, for animal only. But anyway, first one has to take animal ethical clearance before going ahead with this type of experiments. You first obtain that, then go for this.
One more question from in the chat box from the Pooja Yadav. At what location we are injecting the level platelets? Is it directly to the mesenteric or carotid artery, or we can inject them elsewhere in the body? So, label platelets we are injecting always into the intravenous. So, intravenous site either we can choose from the tail vein, and or from the retroorbital injection. So, these label platelets are not giving directly to the mesenteric or carotid artery.
In the next session, we will also show how we injecting that antibody into the mice.
Any more question? Sir, next session.
I think we can now proceed with the next session. More questions will crop up by then. And again, we'll be going for the question answer session at the end of next session. So please proceed with the video.
Very good morning to all of you. In this video, we are going to demonstrate the method section for the intravital microscopy to see the formation of arterial thrombus in ferric chloride induced mesenteric artery of a healthy mice of 4 to 5 week olds weighing 8 to 10 gram. So here in this case we are taking a mice which is of 8 gram.
Yes, sir.
So this is the mice and this is the male mice and it weighs 8.78 gram. So first we have to anesthetize by using the ketamine and xylazine. Ketamine cocktail is 100 mg per kg and xylazine is of 10 mg per kg. And we have to inject 100 microliter of ketamine xylazine cocktail to a 20 gram of mice. So we are calculating by the weight So we just take our volume. Next, we take an insulin syringe, which is of 1 ml, and we aspirate all the anesthesia into this syringe. And first, we remove all the air bubbles. And this mice hold by this way. See, like this. And we have to inject the anesthesia by intraperitoneal. This is the way how to inject. Now we just again put it into the box and wait for 5 to 6 minutes. So we can pause. Pause, okay, sir? So after we inject the anesthesia, we check after 2 minutes if this mice is getting anesthetized or not, fully anesthetized. So we just press on this tail, and then if there is no any movement, then we find this mice is getting anesthetized. And also check with their feet. There is no movement. Again with this, and there is no movement. And next step is we have to check their heart rate. So you can see here there is the beat in their hearts. So mice is alive, and but this is anesthetized, and this mice is getting stable for 2 to 3 hours. So now I am going to start our experiment. So first we have to after anesthesia we have to inject and platelet, because we are going to see our formation of thrombus in arteries. So to detect the thrombus formation by the platelets, we have to label platelet with a specific antibody, which are labeled with the Alexa 488, and which labels platelet specific receptor glycoproteins. So now we are going to inject, and mice weight is 8 gram. So we have to take 1 microliter for the 1 gram of mice. So we will take 8 gram of antibody. अभी सर लाइट बंद करना पड़ेगा सर, स्टॉप 2 मिनट करना पड़ेगा. So this is the antibody. This is the daylight X488, and this bind with the mouse platelet labeling, and this is purchased from the M-Trade. So we'll take 8 microliter of this antibody. So we are taking 8 microliter of this antibody, and we are taking this vial. Sir, let's go. Next step, we have to inject this antibody into the mice vein through the retroorbital injection. So retroorbital injection we perform to inject antibody. First we remove the all air bubble.
नहीं vortex नहीं.
Now
Real current.
How to inject? First, this is the position. We inject with the 45-degree angle. So, Yes. And after injection, we just mesh with their eye. This is done. And then turn right. इसका ये बीट दिख रहा है? बीट थोड़ा सा पास बीट देख लीजिए एक बार सर. माइस इज बीटिंग. किरण सर ठीक है. Then so we take this portion of this mice and we just dissect from longitudinal from this side to this side, and then after the cutting we just remove the skins and expel out the intestine to see the mesenteric arteries, to expose the mesenteric artery. ठीक है सर, इतना ही करता हूँ. So after the dissection, we can see here very clear mesenteric artery here, and this side, right side is the mesenteric artery, and the left side is the veins. So we focus in the microscope this artery to see the formation of thrombus in ferriculoid induced model. नाम बोलना. हाँ, start. So for the imaging, for the imaging we are using the software named NIS element. So here is the NIS element software. We just open it, and you can see here NIS element advanced solution for your imaging world and used in advanced research. So this is the windows for the NIS element software, and you can see here the AVI acquisition panel and DS-FIN setting for the fluorescence enhancement and fluorescence down for a particular image. And we can once we are ready, then we just start with the record.
थोड़ा सा फिर से बताओ, वो ना वो उधर था ना टेढ़ा था.
हाँ.
वो फाइल
कौन सा बताऊँ? VS.
हाँ फिर से.
So this is the NIS element software windows, which is from purchased from the Nikon, and this windows shows the following features. First is the AVI acquisition panel, through which we can record particular videos or image, and we can save it into the particular folder. This is the folder location, and we can mention here the sample name. And this is the DS FI1 setting, and through this setting we can adjust the fluorescence excited or down regulate of a particular site. Like if you can expose the fluorescent 600 millisecond, or we can this is the number of the panel 6 millisecond to 60 second. This is the exposure time for the particular laser light. And this is the laser lamp. This is the laser lamp which provide the exposure to mesenteric artery, and that are visualized in this software. So we just click on the play option here, live, and we can see here. This is the artery, and this is the vein. थोड़ा सा इधर इसको नहीं उसको आप थोड़ा इधर भेजिए, ये पैनल थोड़ा इधर बीच में लाइए. फोकस नहीं सर, बीच में लाइए इधर. हाँ ठीक है. So you can see for this particular mice, this is the artery and this is the vein, and we focus here for the formation of thrombosis, and we just damage their wall with the ferric chloride to induce the formation of thrombus. So next step, we just dip a paper with the we just cut a filter paper, very small filter paper, and we just show you how thin it is. So this much thin is paper, and we just cut it a small piece of this. Yes, and this piece of filter paper was dipped in. This is the filter paper. This is the filter paper, very small, and this filter paper was dipped in the 10% of ferric chloride, and after dip, we just put on that mesenteric arteries at this place, but this particular place. So stop.
No, please. Yeah. Now you can speak. So after the you can see in this video there is an filter paper placed at the wall of at the site of artery, and you can see the punctate structure of which in light green in color are aggregates of platelet is getting formation start where paper is placed. So this filter paper is removed after 3 minutes of exposure, and you can see here is this mouse is yes. No. Yes, our pointer is there. Yes, take it. Take it. So you can see here is the formation of small thrombi is there. So in the intravital microscope, intravital microscopy and data exportation, we first see the formation of first thrombus. So you and that thrombus is of about 20 micrometer in diameter. So you can see this this is the thrombus which is of about 3 micrometer in diameter, and this is the control mice which are vehicle treated, and you can see there is the growth of formation of thrombus. And with time you can see there is increase in the formation of thrombi. So earlier it is of about 20 micron meter, and then 30, and then it is become larger and larger. And after 15 minutes, this you can see there is a huge number of thrombi on both side of artery. And after the 15 minute, you can see there is the blockage and there is an occlusion of thrombi, and through which no platelet is getting moves. And after the control mice, here we show the treated mice. So this is the artery, and right side is the artery and left side is the vein. And in the artery, again we'll induce with the 10% of ferric chloride. And after the induction, so in control mice, we found after 15 minutes there is the complete occlusion, but in case of control mice, we did not has the first formation after the 3 minutes and 5, and we can see here after 10 approximately 10 minutes of injury, we can see the first thrombus formation in case of treated mice. And this thrombi will increase their size very slowly. So after 15 minutes, you can see this is the thrombi which are formed in treated mice, and while after 15 minutes in case of control there is the complete occlusion, but in case of control there is no major thrombus formation is there. So in this mice, we are using anti-thrombotic drug or anti platelet drug for a particular mechanism.
So as you can see, till the 40 minute of till the 40 minute, there is no occlusion of this artery. So we can tell that this drug which are using in this mice act as a antiplatelets, antithrombotic drug. Yes. So is there any questions regarding this demonstration video? So you can ask. Unmute to all. Unmuted, na?
Hello, sir.
Yes, yes, sir.
Just I have a query. And what is this background of this animal?
Background means, sir?
Which strain, whether it's which kind of strain it is?
I'm using Swiss albino mice.
Swiss albino. Okay. And one more query is, you use this ketamine and xylem combination, no, for anesthetizing and all.
Yes, sir. Yes, sir.
So instead of doing with that, can I proceed with isoflurane also?
For isoflurane, sir, we can use, but in isoflurane mostly use in case of laser injury model, because in either if you have the setup for the isoflurane with the oxygen exchange at the setup of your microscope, then you can use isoflurane also.
Okay. Thank you for the explanation. Thank you.
Yes, sir. Yes. And in the chat box, there is question from the Ibenla Hedden. Is the artery punctured? So no, ma'am, there is no punctured artery. It is only injury that we are created by the ferric chloride.
So ferric chloride basically percolates into the artery and causes denudation of the arterial surface. Okay, so that causes the injury. That causes the tissue injury. There is no physical injury which has been done to the artery.
And next question from the Vishnu Priya. Can we use this model in rat also? So in intravital microscopy, we basically prefer the smaller mice that weights is near about 8 to 10 gram. And if you are asking about the week, so it is about the 5-week mice. So why we are taking this smaller mice to avoid the very non-specific fluorescence of antibody due to the presence of extra fat. So if there is a large fat on the surrounding of artery, so our image is not our video or image that we are capture are not good. So that's why we are avoiding for the larger animal to image intravital microscopy. Next question.
Hello, sir. Thank you, sir. Very nice presentation. Sir, I have a question myself.
Please introduce yourself. Yes, sir. Anju Jaiswal, you are asking something.
Yes, sir.
Yes, sir. I have a query. What is the anesthesia dose, and sir, which location it can be taken? It can be given.
So in my lab, we are using the ketamine xylazine cocktail, and ketamine we are using 100 mg per kg, and xylazine we are using 10 mg per kg, and we are using 5 microliter. We are using 100 microliter of this cocktail to inject into 20 gram of the mice. And
okay,
this was injected intraperitoneally.
Intraperitoneal. Okay, sir. Sir, one more question I have. Is there any alternative of ferric chloride?
Yes, repeat your question, please.
Sir, is that have any alternative of ferric chloride instead of ferric chloride
So,
induction?
uh,
In
before this demonstration, you can also see the presentation which are presented by Dr. Paresh sir, and he'll explain the how many way to induce the injury in vivo model. So there are ferric chloride model, laser model, and other agonist model. So there are numerous.
Sir, no, sir. My question is that have any chemical compound that can induce thrombosis instead of red?
Uh, Ferric chloride, actually, I don't have the idea about. And then if you can mail to
Ferric
us, and
chloride
then
is
I'll
basically used because it is really inexpensive and widely available. Also, it has shown its potential in causing thrombus in numerous experiments. So it's a time-tested one. So definitely, we are going to we generally go with ferric chloride.
Okay. Thank you, sir. Thank you so much.
One question in the chat box. Since does it mean we use only very young mice? Yes. Another from another question. Since you said that using larger animal affects the leveling of platelets, so no, ma'am. This larger element does not staining is not affect the platelets, and this is the quality of image that we are focusing in the intravital microscope. So if you are taking larger animal, there is more fat, and that fat will interfere our main target of particular image. So that's why we are not using the larger animal. So this is not about the platelet labeling. How much time is required for antibody incubation? So we are not incubating antibody. This question is from Pooja Yadav, and how question is how much time is required for antibody incubation? So we are not incubating the antibody. We just injected it through the retroorbital injection, and then proceed for the imaging. So no need to incubate. Next question. Suppose we want to study on aged animal, will it be compromised? So, yes, sir. Yes. Can Alexa Fluor 488 fibrinogen can be used for platelet labeling? No, this platelet will labels only activated platelets. This will bind with the platelets that have the change their integrin conformation change. So this particular fluorescent tagged fibrinogen not labels all the platelets. So initially because we are injured, and then check the all the platelets movement. So that's why this we cannot use. Open the all the session. Is there percentage? Any more questions? Yes. Yes.
Hello, sir.
Yes. Yes. Tell me.
Yeah, I have a last question.
Yes,
as
sir.
you mentioned that what are the age of the animals?
We are using 4 to 5 week mice for the intravital microscoping, and for the different in vivo experiment, like if you are using tail bleeding assay, so we are taking 2-month mice, or if you are have for the mice bleeding for to take the blood sample, we are taking aged mice. So this all depends on the different type of experiment.
Yeah. Why? Because these disorder basically associated with aging under, no? So,
Yes.
So age is the culprit for your thinking in that context.
So we are taking only healthy mice, not the healthy mice for the control and treated, and we can check by the assessed by the veterinarian, which assess that this mice is healthy or not. So in healthy mice, we are only proceed our experiment.
Yeah. Thank you.
One more question. Chat box is.
Hello, this is Dr. Ibanala from A.M.S. Kalyani.
Yes, yes, ma'am.
Yes, So actually, I mean, since you said like, you use very young mice, then if we use the, aged animals, will we get good results or something? Because there will be more fat deposits and all that. What is your take on it? Can be used like for aging studies, like healthy aged mice?
You can use healthy mice also, but I think model is different. If you can thrombus embolism model, you can use for the aged mice. Thromboembolism is another way to see the formation of the thrombus in aged in drug treated or control mice. So for the intravital imaging you can use, but the quality of your image is not much good. That's why I can suggest this was not for the aged mice.
Okay, and the microscope is like the inverted fluorescence microscope, right? It's like
Yes, we are using inverted microscope.
okay. Okay, thank you.
Yes, and one question is there from the Priyanka Das to everyone. Is 8 microliter amount of antibody is fixed for certain mice, or it's varies? 8 microliter amount of antibody. No, this was not fixed. We are 8 microliter administering. So if you are 1 microliter per gram of mice, we are injecting the antibody. So if mice weight is about 10 gram, so we are taking 10 microliter of this antibody. So this is the very small volume for the injection. So we are not directly injected this volume. So this was further diluted with the PBS, and in the PBS we are adding 8 microliter or according to the weight of mice 10 microliter or 12 microliter, and that with the diluted with PBS and injected finally into the mice. The mice remain alive after doing. Question is from Pooja Yadav. Where is it? Niche. Yes. Does these mice remain alive after doing this intravital microscopy experiment? So during the course of experiment, the mice is alive, and after the complete experiment, we because this mice is injected with the drug and exposed with the different antibody वगैरह. So finally, we euthanize these mice by the cervical dislocation.
So I think we'll conclude it here today. And let me take the opportunity to welcome Dr. Prasenjit Gushai sir. He is an eminent scientist from Regional Center of Biotechnology, NCR Biotech Region Science Cluster, Faridabad. And sir has now been online. And he had previously professor at since 2016, he is professor at Regional Center of Biotechnology, Faridabad. Previously, he was associate and assistant professor, in the same lab from 2011 to 2016. He is also assistant professor at Baylor College of Medicine, Houston, USA, from 2008 to 2011. He has membership of many professional and academic bodies, including Academic Committee of ESIC, Reviewer of US-India Fulbright Nehru Scholarship, Reviewer of India-Australia Biotechnology Fund, and many others. So the list is unending. Sir has also selected publication in clinical and translational medicine, and almost 16 publication in many important journals in national and international.
Stop sharing.
And let me welcome sir here. And sir will be basically focusing on many important topics, but the main area he will be focusing here is effect of thrombosis by COVID-19. So this has been a very recent issue, and many of us are facing problems with the long COVID and other circumstances. So definitely, sir's address here will enlighten us on this current and important issue. So I welcome Dr. Prasenjit sir for his talk. Welcome, sir.
So thank you very much. Good morning to everyone. Can you hear me?
Yes, sir. You are very much audible.
So my slide is visible?
Yes, sir. Your slide is visible, sir.
Oh, great. Thank you. So I would like to thank the organizers and to invite me into this. I think this is the country's first center. Can I just a minute? Can I have this laser? How to get it?
फुल स्क्रीन करो और मेरा हटा दो.
Just give me a minute. I just try to.
Yes, sir. Yes. Sir को लाओ बीच में.
Okay, good. Thank you. So, yeah, I think this is the country's first center which is created by ICMR. This is a platelet research center headed by Professor Devabrata Das. And I think he did a wonderful work. And that's why ICMR created this center in your I.M.S. B.H.U. And I always feel proud of being a part of B.H.U because I did my PhD there. And so thank you very much to the organizers, and thank you to Professor Devabrata Das. So today I'll be talking about this platelet activation and thrombosis in COVID-19 and molecular signaling to therapeutics. So you know that when I just first pulled up that the literature, number of literature published till date it's on COVID-19 and thrombosis is 5,400. So it tells actually that how active the researchers are, and humongous amount of good work has been done during last 2-3 years. So one of the things I also feel proud to be the part of this research community, thinking that, you know, that such a pandemic has been, that issue has been solved quickly because the proactiveness of the researcher and the clinicians together. I think they are the main component of this solution, which actually brought to the world so quickly. Now there are more than hundreds vaccine reputed companies vaccine against this COVID-19. So because this is the thing, and if you remember, within the pandemic started, first mRNA vaccine came within just a few months. So this actually tells that how that research and the techniques, everything developed so rapidly. If you compare with another such pandemics, viral pandemics, is that Spanish flu virus, which was in 1918, and the first vaccine came in 1942. But in this case, if you compare that with this COVID-19, the vaccine, it's so this is because of that, as I said, the contribution of the researcher and the clinicians ultimately won the match. So that's why I feel part of this scientific community. And what actually we did, as I said, that so can you hear me? Am I audible? Am I audible to you? Okay. So okay. So you can. So that so these are the works we did actually. That if you see this, just I'll focus on this. How what is the contribution from our lab? So we can tell you that into these things. So when there was a huge demand for all these things, what we did actually, we started working on this COVID-19 pathophysiology, and we have the animal model and all these things, and we started working and published this paper consecutively. And what actually we have shown that one of the metabolite of the Krebs cycle, since you people are from the biochemistry department, you understand the Krebs cycle metabolites, one of that alpha ketoglutarate. So alpha ketoglutarate, we actually used as a therapeutics, and I'll tell why. And we found it actually rescued the SARS infected animals dramatically. So the reason being is that, so here few things I want to introduce to you is that alpha ketoglutarate, prolyl hydroxylase 2, this is an enzyme which actually is this is an enzyme, and the substrate for this enzyme is hypoxia inducible factor HIF1 alpha and that phosphorylated AKT. So HIF1 alpha and phosphorylated AKT are the two major substrate for this enzyme. So what we did actually, if you look at this one, what it tells that this enzyme is an oxygen and alpha ketoglutarate dependent enzyme. So for its activity, it's used. Both the things are needed: oxygen and alpha ketoglutarate. So when it is functional, so what is the function of this PhD2 major function? It's actually cleave the proline residues of its substrate. So two substrates, one is that HIF1 alpha or HIF2 alpha and the phosphorylated AKT. It cleaves the proline residues of this substrate and inactivate this function of this substrate. As you know, that HIF1 alpha controls more than 300 genes in our body, and phospho-AKT, I think it's hugely important. But what we are trying to show here, that when we treated the mice with alpha ketoglutarate, it actually inhibits that the platelet activation, because platelet activation is mediated by phospho-AKT. And because, as I said, alpha ketoglutarate, when in presence of alpha ketoglutarate, PhD2 enzyme is activated, and it is under normoxic condition. Normoxic means when oxygen is available. So in presence of oxygen, in presence of oxygen and alpha ketoglutarate, PhD2 is active, and its hydrolase and cleaves inactivate that phospho-AKT and HIF1 alpha by proteasomal degradation. So this is the things, is the main concept behind these things. So how it is important? These are the person Nishit and this Sakshi and Simran. They are the major player behind this publication. So as you see, this is the animal model. You can see that this is the injury in the lungs of the SARS-CoV infected hamster. You see that red patches, everything is a mark of injury, and it happens actually in the human being also. So when the COVID-19 infects, it's damaged the lung very severely. So that most of the people died because of this. What happens actually, that it actually blocks the alveoli. So there is a huge portion is damaged. Oxygen passing through the alveoli to the blood, each being inhibited. So that's why that condition is created, which is called hypoxemia, which is that decrease in oxygen concentration in the blood. So these are the reasons that lungs is so hugely damaged. So that's that oxygen passing from the air to blood, it's actually it's limited by this infection of this SARS-CoV-2. So this is one of the major reasons. Even those who survived, what happens after survival? If the lung is damaged and it was not properly treated at the time of infection, so these lungs nowadays actually after this COVID, when this pandemic is gone, now the main focus of the researcher is how that this damaged lung, actually now research is focused on that this damaged lung, most of the time actually what happens, it actually transforms into fibrotic lung. So fibrosis happens and all these things. So totally damaged lung, even the people are surviving, they're unable to have normal oxygen saturation in their blood, and their lungs is so limited, they cannot do the normal breathing and all these things. So they need still oxygen. And I have one employee of RCB, still I know, so she is the survivor from Delta SARS-CoV, and still she needs every day, she is working for some time, and still every day she needs infrequently in 2-3 hours interval, she needs oxygen. So this happen. So that's the things needed during infection. The proper treatment is needed to rescue the lung. So what I said, when we fed these animals, which is SARS-CoV-2 infected with the alpha-ketoglutarate, just this is a timeline happens is that 5 days in case of hamster, or we have two models. One is the hamster, was the best model. Another is that we took that Balchi mice and intranasally we just gave adenovirus construct of human SCE2, and so they transiently express the SCE2 in their nasal passages. Then we infected, because for this virus you need SCE2 is one of this is the main receptor for this virus. So SCE2 when transiently expressed, we give the virus infection nasally, intranasally, and in from the second or the second days, they start showing symptoms. They are sick, they cannot eat properly, they cannot drink properly, and all these things happens. And by the day 5, 6, they are sick, very much sick, and after that they recover. So these are the two models: transiently expressed SCE2 model of mice and hamster. They generally do not die. But if we give too much virus and all these things, they sometimes die. But otherwise, they don't die. On the other hand, there is that stable transfected SE2 transgenic mice. They get severely. That model also we are started working on that, but that might simply die in day 5, because their SE2 is so highly expressed that when we give this virus, they just get sick and they die in day 5 or 7. So, but we used, as I said, the transiently expressed mice model, SE2 mice model, and the hamster. So this is the things, as you see, these are the clots of the that inflammatory clots, means in cytokines, platelets, everything is together. This is the thromboinflammatory complication, which is actually damage the lungs. So in this paper, what we have shown actually, so just I put this again, this cartoon that for your refresh your memory, that what we did actually. So we first tested that in general, what happens, that what we found that it's just if you forget for a few seconds SARS-CoV, what happens? So we use this alpha-ketoglutarate to test that. So first we checked that whether in normal condition platelets and monocytes being regulated by this mechanism, when we supplement with alpha-ketoglutarate, isolated platelet and monocytes in vitro. So if we supplement with alpha-ketoglutarate, what happens? So actually what we found that PhD2 activity increases and it decreases that. I'll show you the data. So it decreases the phosphorylated AKT, and that's how platelet activation is decreased. So this is when we give alpha-ketoglutarate. It augment that increase the activity of PhD2 and decrease the phospho-AKT. So on the other hand, if we give DKG, which is a stimulator, which is an agonist of this. Sorry, it's an inhibitor of this PhD2. What happens actually that it reduces the PhD2 activity, and that's why that phosphorylated AKT increases. So if you treat anything with DKG, ultimately phosphorylation increases. If you treat with alpha-ketoglutarate, phosphorylation decreases. So phosphorylation decreases is related to less platelet activation aggregation and monocyte activation, leukocyte activation. If you give alpha-ketoglutarate, if you give DKG, just opposite things you observe. So we also tested in the mice model. I'll tell you what we did and all these things. And so this is the data. You see that. So we just took normal biopsy mice and fed them with 1% alpha-ketoglutarate for just 48 hours and all these things. And then isolated their blood and did the platelet aggregation. As you see, their platelet aggregation is highly, I think before that I have another slide. Okay, so okay, so this is actually this slide has to anyway. So this platelet aggregation, so what happens actually that mice fed with alpha-ketoglutarate, so they have decreased platelet aggregation. And this is another mice model we created, carrageenan mice model. So that mice treated with carrageenan and fed with alpha-ketoglutarate, as you see this is one of the things that's showing that microthrombus formation in the lung and in the liver in the carrageen treated mice. When this, and the mice carrageen treated but supplemented with alpha-ketoglutarate 24 hours before the carrageenan treatment or 48 hours before the carrageenan treatment, their lungs are pretty much cured because of this less thrombosis and inflammation. We measure all this. These are the bars showing these things. And this is the tail thrombosis we measure in the mice. So this is, Professor Das has very fascinated instrument that I think before my talk you have been showing that intravital microscopy. If you see under the intravital microscopy, you will see that alpha-ketoglutarate, dietary alpha-ketoglutarate decreases the thrombus formation. So this is the thing. And what is the molecular basis behind it? If you see, this is for the first time we have shown that platelet have PhD2. Platelet has different subtypes of different subtypes of hypoxia inducible factor that has been shown by Professor Das for the first time in platelet. But we have shown here that platelet has all three subtypes of PhD2. And what we did actually, if you look at this data, we activated platelet with collagen. When platelets are activated with collagen, so you see that phospho-AKT is being highly elevated. When we treat it with alpha-ketoglutarate, so you see that phospho-AKT is decreased. As I said, when you inhibit, so this is because this is through that PhD2. I'll tell you how. And this is actually that when you treat with, when you supplement with inhibit that PhD2 with DKG, one of the inhibitor, you see that phospho-AKT is back again. So this is how that it's happened. And what you see here, actually, we pull down that phospho-AKT activated platelet with and without alpha-KG treatment. And so just a minute. So can you just talk little like? Sorry about that. When what we treated that platelet with collagen and collagen plus alpha ketoglutarate, and then we pull down the phospho-AKT, pull down the lysate with phospho-AKT and blot it for this PSD2, and is that hydroxyproline. As you see that. So this is actually showing that this actually, as you see that actually it's so the association of this hydroxyproline and the PSD2, it's high when these platelets are inhibited, phospho-AKT is inhibited. It's showing that what I said, PSD2 clips the proline residues of this substrate, phospho-AKT. So that's actually showing the association between PSD2 and that proline hydroxylase. What I am trying to tell you that when platelets are activated, so there is less the PSD2 activity. When platelets are deactivated, that means there is increased activity of PSD2. It's an inverse correlation between the PSD2 and the phospho-AKT. So we did this in vitro study showing that PSD2 inhibits that platelet aggregation. We did that parallel flow chamber assay where we coated the plates with collagen and perfused to whole blood in presence of DKG and DKG plus alpha ketoglutarate. As you see that DKG is that as you see, pro phospho-AKT. So when we added alpha ketoglutarate, that platelets became inactive, and as you see, the thrombus formation also diminished. So that's how what we are trying to tell. We use this concept. So still that there is no story of COVID-19. I'll tell you that this is now we are convinced that it works through this. So this pathway works like this under normoxic condition. If you give, there is enough oxygen. PSD2 is an oxygen-dependent enzyme. But when you add more cofactor like alpha ketoglutarate, they get activated and they just decrease the function of HIP1 alpha, one of the substrate, and another substrate decrease the function of phosphorylated AKT. Not only AKT, phosphorylated AKT. So this is how is that we use this concept in that the SARS-CoV, as I have shown that in SARS-CoV infection, inflammation and thrombosis, one of the major reasons for this lung inflammation and lungs function impairment, everything. As you see that in these things, we use that hamster as a model. We did also in the mice, but I'm not showing you here these things. So when the hamster was treated with alpha ketoglutarate, as you see that the infection decreased. But more importantly, that infection viral load of the SARS-CoV decreased. But that is different issue. Issue is here that if you see that SARS-CoV actually infected animal, as you see that this is a normal hamster lung, and so when they are infected with SARS-CoV, it is a huge amount of inflammation. This is inflammatory immune cells accumulated in the lung, and huge elevated amount of IL-6, TNF-alpha, IL-1 beta, all the inflammatory cytokines are increased, and in conjunction, you see that microthrombi, these are the microthrombi in the lung increased. If you compare, if you remember that picture that I have seen, I have shown you that lung is damaged, even it is visible from the outside. So these are the happens inside the lung. So, but when we treat it with alpha-ketoglutarate, there is a significant increase in the decrease in the inflammation and thrombosis. More importantly, if you see that apoptosis, that SARS-CoV, that apoptotic cell damage, it's hugely increased in SARS-CoV infected animal. But when they are fed with, supplemented with alpha-ketoglutarate, as you see that significant rescue in the apoptosis in the lung. So this is how, what I told you the story, that lung damage can be rescued by supplementing alpha-ketoglutarate, and this alpha-ketoglutarate has been used for a human trial by several investigation and all these things. This is kind of safe because it's our metabolite, but there is a limitation. Alpha-ketoglutarate has many other targets also, like it targets histone demethylase and so many things. But if you keep that in a focus that you want to rescue risk versus benefit, if you assess. So when somebody is dying because of this thrombosis, inflammation, this apoptosis, so you can treat the mice with alpha-ketoglutarate, and there is, and also I'll show you the later slide. There is less side effect. It doesn't mean anything that why we should not give these things, because and this is doable. And as I said, it has been used for human trial. So that's confidently we used it. So these are the graphs showing that the parameters, how when we gave alpha-ketoglutarate, how these symptoms are decreased and all these things. So as I said, ultimately what happens, I told you that lung inflammation increases in lung inflammation, thrombosis limits the oxygen passing from the air to the blood. So that is the main function of the alveoli. So those are blocked, that are damaged, so many things. When we treat it with the, and this is the oxygen level we measure in the hamster. And when we get, this is the red line, is that SARS-CoV-2 treated animals. When we treated with SARS-CoV-2, that oxygen level hugely decreased in the animals. But when we supplemented with the green dot, supplemented with alpha-ketoglutarate, that oxygen level started increasing. So this is actually showing that there are so many limitations. We couldn't do too much, put too much animals, so many things. All the experiment was done under the BSL-3 facility. So nothing can be out of BSL-3. So there was a huge limitation of doing experiment and all these things. But what we could do actually, we could convince you that when you rescue that inflammation and thrombosis and apoptosis in the lung, you can restore the normal oxygen saturation in the blood. So as I said, there are minimum, so now when you are giving alpha-ketoglutarate, so one thing is that in virus infected animal, main thing is that does it interfere with the antibody production against SARS-CoV-2, because that is the main point of inhibiting that any viral infection, because when virus infect, body start making antibody to neutralize that virus. So that is one of the major defense mechanism of the body. So that's why people gives vaccine to alert your immune cells like B cells, T cells, to memorize the antigen. That is the vaccine technology, right? To memorize the antigen. When real antigen attack the host, so those T cell and B cell start fighting against it and producing antibody and neutralize that virus load. So that's how we rescue from our virus infection. So we ask these things, whether alpha-ketoglutarate interferes with that neutralizing antibody production by the host or not? And the answer is no. When we give alpha-ketoglutarate, there is no significant effect of, so this is that antibody produced against the, these are the antibody levels against the SARS-CoV-2 in infected animal, and this is day 5 and this is day 9. So as I said, when we give alpha-ketoglutarate, there is no significant difference in this production of the neutralizing antibody level in the animal. So it is safe. So now the thing is, also we measured that that neutralizing capacity of the, this is the antibody level in the specific antibody against SARS-CoV level in the plasma. So then we use this plasma to neutralize these virus ex vivo. We collected the plasma from a alpha-ketoglutarate treated SARS-CoV-2 treated SARS-CoV-2 infected alpha-ketoglutarate treated animals and only SARS-CoV-2 infected animals, and did the neutralizing assay in ex vivo. And we've also found the neutralizing antibody level in the plasma of both the subtypes. There is no significant difference. Means what we are trying to tell, alpha-ketoglutarate doesn't affect the that adaptive immune response of the host. That is the main concern is here. So, that's why we just have shown this in this cartoon, that when there is damaged lung, and if you give alpha-ketoglutarate through this PhD2 AKT axis, rescue that inflammation and restore that oxygen saturation in our body. So this is with this concept. With this concept, we are doing several other projects right currently. As I said, that now the major concern is that when damaged lung started growing again, so what happens? Many of the damaged epithelial cells and epithelial to mesenchymal which is called AMT, that when the fibrosis happens or cell damaged, so before that these things happens, that epithelial cells transform into that mesenchymal cell, which is called epithelial to mesenchymal transition, AMT. So what we are trying to understand now, that how this lung fibrosis, what is the severity of lung fibrosis in these models, and whether it can be rescued by alpha-ketoglutarate and other anti-phospho-AKT antagonist and all these things. So we are trying to understand the pathophysiology of ARDS, because this is a complete acute respiratory distress syndrome. How this ARDS and that fibrosis, it's actually creating the problem in this infection. Now we have model of different models. So now also we created that diabetic mice model. And as you know that it is known that when the diabetic phenotypes, diabetic people are mostly affected by this SARS-CoV infection, because of diabetic people have less T cell response, and the immune response is very much compromised in those diabetic patients. One of the main reason. But we have now mice model of diabetes, and we are infecting them with the SARS-CoV-2 and trying to understand this ARDS pathophysiology and this fibrosis pathophysiology. And I'm not showing the data, but our data is showing that diabetic pathophysiology, mice having diabetic pathophysiology are more vulnerable to this infection, and their lung damage is hugely compromised. Their ARDS, we measure some of the component of that marker for the ARDS, and also that fibrosis marker. And we have seen that hugely these are increased in diabetic, when the diabetic mice is infected with SARS-CoV-2. So what we are trying to address this problem using different mice model of these things, and trying to understand whether it can be rescued by this alpha-ketoglutarate supplementation or not. So this is one of the things we are doing in this mice. And also we are trying to understand the platelet pathophysiology and pathophysiology of the immune cells and all these things, as I said. So besides that, we did another work during the COVID. We collected, we wanted to understand that platelet. We collected samples from the COVID patients with mild and severe. So with COVID patient during the first infection, not the Delta, before Delta, the first infection, we collected the, so this Simran actually did this work. So we collected these samples from COVID patients. And the platelet, we just, we analyzed, we collected platelet, isolated, and analyzed for whole transcriptome of the platelets. So how do you do? So we collected asymptomatic and symptomatic patients from the ICU. These are the red dots, are the red dots which platelet has been used for transcriptome analysis. But this is all together is that, as you saw, that based on the D-dimer level in the plasma, so we segregated that patients, and all the ICU patients had high D-dimer level, and based on that, we thought they are highly thrombotic coagulative problem also they had, and their MRI reports also shows like that, and all these things. We collected these red dots platelets, and the platelets from this healthy individuals. We didn't include this when we did that transcriptome analysis. We just used these red dot platelets from the ICU patients, because there is a limitation which is cost about lakhs of money. So and we used that healthy individual's platelet, and we did this transcriptome analysis, and I'll tell you in the next slide. But if you see that thrombin level was increased in this level, other parameters we could measure, we measure that platelet microparticle, which is a marker of the platelet activation. We couldn't measure platelet activation level because of some technical problem during that time. But using these things and also the platelet leukocyte aggregates were increased. So these are the markers we could measure and show that. So there are highly thrombocoagulative problem these patients had. And then we did this, analyze the transcriptome data. As you see this, sorry, I'm sorry. So I used that asymptomatic patients platelet and that ICU severe patients. So these are the 9 severe patients, and asymptomatic patients platelet we gave in both the sites. As you see that this actually tells you that profiles is so distinctly different. That transcriptome profiles of these, that severe versus asymptomatic is so nicely aligned in those things. So we did huge analysis and all these things, and then it's a bucket of 100,000s of genes. You need to, then we started fishing out which is needed and necessary for our work, and with their score and everything. As you see that, this actually tells you that the coagulation pathways, these are the pathway analysis, coagulation pathways, TNF signaling pathway, IL-6 pathway, all these things are highly elevated, P38 MAP kinase pathway, all are highly elevated in these severe patients compared to these asymptomatic patients. So these are the sting analysis is showing that measure molecules of these pathways, how they are interconnected, and all these things. So with these things, so now the question is that what happens? That we found that elevated level of IL-6 and TNF alpha signaling pathway in platelet. So now the question that, and it is known that IL-6 and TNF alpha both can activate platelets. So, and it is known that IL-6 and TNF alpha is highly elevated in these patients because of cytokine storm, so many things. So we measure these things, that whether these individuals has elevated level of IL-6 and TNF alpha in their plasma. And this is actually, this data is showing they had, again, so red dots are the dots which platelet has been used for that RNA-seq analysis. So this is, as you see, that IL-6 level and all these things are highly elevated in these patients. And these are the platelets we could showing that P60, P38. Since we have seen this elevated level of those pathways, P38, ARC, and all these things, we measured these things in that platelet pellet we saved. And as you see that this ICU means severe patient has elevated level of phospho-38, phospho-ARC, and all these things. So these are the densitometry analysis and all these things. So that's why we've just narrated in this cartoon showing that elevated IL-6 and TNF alpha can activate platelets because their IL-6 and TNF alpha signaling, P38 signaling pathways or MAP kinase pathways are elevated, and which actually results in that elevation of this blood coagulation and thrombosis pathways, and that's how it's activate platelets, and it's actually, that's why it's become more thrombocoagulative and all these things. So this is how it happens. So this is the stories we could contribute, but as I said, many ongoing works are on my lab is going on, and Professor Das is aware about it because I share my many of the ongoing works with him and all these things. Just this is for your knowledge that what we are doing and all these things. As I said that, so this is a dangerous disease, and but I'm so happy to tell you that within 2 years, total world was upside down. But now that endless effort of the researchers to make vaccine and the doctors' treatment that brought the world again in the set. So this is because of this researcher and the clinician. Nobody else, yes, it's a collective work, but major contribution, if you think that researcher and the clinicians work together to just to brought this world in normal set. So this is with those thanking to the researcher and the clinician. That I want to acknowledge few of the students, Simran, Neset, Sakshi, and the collaborators like Professor Pandey, Shuman, Nikhil from ESIC Hospital, where we collected the samples. Still, we have collaboration going on. ESIC Hospital in Faridabad is just next to our, not next, 2 kilometers from our institute. So Joe Prakal from Utah for his contribution in one of the work, and Milan Tripti Shankar from THST, our neighbor institute in this science cluster, and Professor Brati is one of the virologists from RCB who just provided virus and other helps. So these are the funding from for which I use for this work, and thank you very much.
Thank you, Professor Guchayat. So it was quite very interesting passage into how hypoxia linking with COVID, linking with lung fibrosis, linking to platelet activation. And I know that for everybody, I can tell that Professor Guchait is doing very robust work on this alpha ketoglutarate, which is a TCA cycle intermediate. So it has such a healing role. And he has, in many occasions, he has showed in many ways, he has validated that it can be a wonder drug, wonder healing drug. It looks very simple molecule, but it has those effects, and due to its action on PHD2 prolyl hydroxylase. So it shows for all the audience today, we just listen to another aspect of platelet activation and thrombosis, that how hypoxia can be linked to platelet activation. So he has linked that whenever there is hypoxia, it links to AKT phosphorylation that one can do by Western, and AKT phosphorylation further is linked to hyperactivity of platelets. So by giving alpha ketoglutarate, prolyl hydroxylase was activated. Once prolyl hydroxylase activated, hypoxia inducing factor is reduced, its activity is reduced, they are reciprocally related. Then AKT is reduced, phosphorylation of AKT is less expressed. So this is I have following his work, and it is a very robust study, and also perhaps he did not tell that he is the person who goes so many times to high altitudes, to Leh and Ladakh, to study why the islanders, the people who are residents of such a high altitude, they are quite immune to such thrombotic disorders. These disorders don't occur in their case. But if we, from the lowlanders, if we go to high altitude, we shall face some consequences, bad consequences. So he has found some gain of function gene mutation in the PHD2 gene. So that is coming out very greatly as a very great discovery. So with this, I thanks from my heart, Professor Guchayat, for giving time. And if you just have some time, that we can take some of the questions from the audience. If,
yes. Thank you, Professor Das. Yes, please.
Yeah.
Hello, sir. Am I audible?
So please introduce yourself to the speaker, and then please ask your question.
Yes, sir. Thank you so much for elaborated talk. This is Mahesh from University of Hyderabad. Hello,
yes,
sir. Yes, sir. I have some couple of questions regarding this animal model. You selected hamster is the only for disease-induced model instead of monkeys and all. So, what is the rationality? First, I want to know that. And
no, I said that I used also transiently expressed AC2 expressing mice model. I didn't show that data. Both are explained in my that publication that I have shown, that we used mice. I have shown you that only hamster.
And one more question is, and you said that hypoxic environment basically is generated by the virus, or itself it is through tissue injury kind of thing?
Hypoxia, since any of the respiratory virus actually cause this hypoxic problem, because the hypoxia is the nothing that when oxygen passing through the alveoli to the blood is limited. So it is reported, this kind of condition also reported for other coronavirus, other respiratory virus, rhinovirus, and other pneumonia virus also. So it's actually a problem of the respiratory virus, and it's not only that, but in this case it is more severely damaged. As I said, when lung is damaged, they are not able to pass the oxygen properly. So that's why automatically hypoxic situation is generated. And it's a very unique thing is that crosstalk between hypoxia and inflammation. Local inflammation cause hypoxia. When hypoxic molecule are elevated, they also trigger the inflammation. So it's a crosstalk between hypoxia and the inflammation. So that actually play a major role over here. Yeah.
Okay. And continuous on that. And you mentioned that inflammation and that is the activation of pro-inflammation and the anti-inflammation. So actually, I didn't see NF-kappa B activity over there. So what is your comment on NF-kappa B activity?
Yeah, NF-kappa B is a huge, it has huge contribution it's a NF-kappa B is inhibited, right? As I have shown, NF-kappa B is a pro-inflammatory signaling molecule. So it has been elevated and it was elevated. I didn't show you the data, but we measure all these things. So and that during the SARS-CoV infection, that NF-kappa B P68 subunit of NF-kappa B, so that was highly phosphorylated. So that I didn't show you, but it is elevated and yeah.
Okay. And my last question is, sir, as you mentioned that overall the study, you mentioned that dietary intervention, can cure this SARS-CoV and all. So, alpha ketoglutarate. And what about the control diet of that animals? So you are thinking that you don't have any alpha ketoglutarate component in the total food and all?
No, I didn't get your question. What?
No, you mentioned that dietary intervention can lead to these viral infections, no? Alpha ketoglutarate through alpha ketoglutarate activity.
Yeah, just we gave dietary alpha ketoglutarate.
Yeah, and what about the control diet in that? Control diet animals.
Yeah, so that's actually we were asked is we had to give that some sugar pills supplement, but we just gave in that because alpha ketoglutarate it's dissolved in water. Just we gave the water, and reviewer asked these questions, but you see, as I said, we couldn't do that during that time. A repetition of these things was not possible, but the valid thing was proper control would have been that if you just give some sugar or something else, which is so.
Yeah. Thank you so much. Nice explanation. Thank you so much.
Hello, sir. This is Iban Lahdem from I.M.S. Kalyani. Sir, my question is, we do that dietary intervention 2 days before infecting the animal. So is it, will we get similar results if we get the animal infected first, and then after that we do dietary intervention?
Yeah, we did that.
No, you did 2 days prior to infection.
No, that's the, as I said, that was the published, but nowadays we are infecting the mice, and along with we are giving this supplement, because this is logical, right? When I get to know that this patient has this SARS-CoV, then only we'll give this supplement. So that's we are doing. What we are doing, nowadays we are infecting the mice with virus, as well as we're giving the dietary alpha ketoglutarate, and it is kind of similar results. So
you give it simultaneously, or after the mice are showing symptoms or something like that?
No, we see that when we give infection to the mice in the morning, in the afternoon we give this gavats, alpha ketoglutarate. Not immediately we give, because after this viral infection we give them 12 hours rest, and then we give this alpha ketoglutarate. So that's worked pretty similarly. What I've shown the results, whether you give before or after, it's kind of similar results.
Okay, thank you, sir.
Yeah. So thank you, Professor Das. I think that.
May I ask one more question? Just
yes, sir.
If you give some precursors of alpha ketoglutarate like glutamate, which is converted to alpha ketoglutarate transaminase, or alpha ketoglutarate formed from what? Isocitrate?
Sir, isocitrate.
Alpha ketoglutarate also is formed from isocitrate. So all these precursors also should work there.
Means we didn't try, but theoretically it should work. But I don't know, because we just used that direct cofactor of PhD2. That we didn't use the we didn't try the precursor of alpha ketoglutarate, and since it is alpha ketoglutarate is available as a dietary supplement, we just simply gave these things. But we didn't try. But theoretically, it should work.
It should work. Yes. Yes.
Sir, have you tried? One of our colleague has a question whether you have tried only oral dose or you have also tried the intravenous ones. And if
No,
you have
oral dose. This
yeah.
is oral.
Acha. So will any intravenous intervention will be more effective or less effective? What is your say on this one?
Since it is that this metabolites is taken through dietary supplements, so we use the diet because which is non-invasive methods. We didn't try.
Okay, thank you, and thank you for your time, Professor Guchai. Now you can be here online, but we shall go ahead with our protocol and plan. Okay.
Thank you very much, sir. Thank you.
You can be there online, no problem.
Okay, thank you. Thank you.
Oh, nice to meet
you. Next, we'll be proceeding with thromboelastogram. First, we'll be going for the theory and the lecture, and then we'll be going for the practical session. That will be again like virtual lab experience. But as we are all of us are here since morning 10, so we'll be heading towards one short break of 10 minutes, and then we'll be heading towards the session theory and practical both. It will be conducted by our lab colleague, Mohammad Iqlaq. Both of them will be there. Okay. So you may go offline for a few time, but please be again sharp at 12:40. Okay. Thank you. के बाद
Good afternoon, sir. It's Shwetanka Prasad from Manipal Tata Medical College. I'm not audible, sir. Means you are not audible.
We are visible and audible. Good afternoon, all of you.
Good afternoon, sir.
Thank you for all for your patience. And now we'll be commencing with the second session of this day 3, and that is application of thromboelastography in characterization of viscoelasticity of the clot. As we know that till this session, we were mainly talking about the action of platelets, but the platelet not only comprises of the clot not only comprises of the platelets, but also with the protein part. So it will be very efficient for the research if we can model our experiment on the protein part as well. And thromboelastography is the technique which we look for the protein part and its function. So, Mohammad Iqlaq, our PhD scholar in the Center of Advanced Research on Platelet Signal and Thrombus Biology in the Department of Biochemistry I.M.S. B.H.U, is here to present. His face is well known because most of the experiments that were performed for videography from the day one, you are witnessing him performing those experiments, and he is our PhD SRF. He has area of research in platelet biology, thrombosis, homeostasis, and signal transduction. The current research he is doing is in the cell death pathway in agonist stimulated platelets and metabolic study in agonist stimulated platelets. Till now, he has 3 very good impact factor publication in eLife, in blood advances, and in hematologica, which span his career. He is also awarded senior SRF and JRF by CSIR, Government of India. He is well versed with the technique of flow cytometry, fluorescent microscopy, confocal, bioflux, Western blotting, and others. And he will be our presenter for this seminar. I welcome Mohammad Iqlaq for his presentation. Over to you.
Thank you so much, sir. So, hello everyone, good afternoon. So, today I am going to present a lecture on application of thromboelastography in the characterization of viscosity of clot. This not movable.
Click on it. Click on it.
Yes. Not working. Yes. Okay. So today I am going to explain about the thromboelastography. So in this slide you can see here is an instrument in left side corner, and this instrument is named thromboelastography. And this, what is the main goal of this instrument? So you can see in the right side corner there is an peg instrument which is shaped in the triangle, and upper of this is the thrombosis and the bleeding. So this peg maintain the blood in a normal state. This tag will help us to find out the particular person blood is getting thrombotic or in more bleeding. So for this purpose, we can assay and we can know the what are the problems in that particular patient. Is their platelets is getting thrombotic, or is blood circulation or clotting cascade is getting toward the bleeding. So by this assay system, we can assess the thrombosis parameter and the hemostasis parameter. And this technique was developed by Dr. Helmut Harter in 1948 at the University of Heidelberg. So in this slide, I'm going to explain what is the thromboelastography. So thromboelastography is a dynamic method of testing the efficiency of blood coagulation. So here we mentioned that it is the dynamic method. It is a dynamic method or efficiency of blood coagulation. So what is the meaning of efficiency here? So it basically assess the interaction of clotting cascades and platelets interaction. So that's why we are mentioning here the efficiency of blood coagulation, that it is the dynamic method which assess the interaction in between platelets and other clotting cascade like clotting factors that are involved in the whole hemostasis or in thrombotic parameters. So this method used by many clinicians are also used in research purpose. So which type of clinician use this method? Like you can see in the next paragraph, it is used in the surgery. Surgery department has used this tag instrument, anesthesiology, emergency, and in other clinical cases that in which the clinician use for the to know the actual state of blood. It is our hemostatic, or it is a bleeding state, or it is in thrombotic state. So next is thromboelastogram. So what are the result? What are the graph that we are obtained by the thromboelastography? It is called thromboelastogram. So we mentioned thromboelastogram is a graphical image of recorded amplitude of the movement of the pin as a function of time. So with increasing the time, this thromboelastograph measures the graph is the hemostatic assay. Voice is clear or not clear? Okay. So in this graph, in this slide, you can see. So what are the steps are involved in the formation of a clot when a vessels is getting injured? So in this slide, we inform how the platelet is getting aggregated at a particular injury site, and how this system is tracked by the thromboelastography. So you can see in the here is an damaged blood vessels. At the site of damaged blood vessel, there is release of clotting factor, and that clotting factor finally help in the production of fibrin. So this first time fibrin synthesis and first time fibrin polymerization is recorded by the R time. R is the reaction time, or reaction time. So reaction time here you can show by the symbol R, and then K is here. K is means how fibrin is getting polymerized. So with how polymerized and have a 20 millimeter of diameter. So this will represented by the K. And next is how fibrin is accumulated. This is by the alpha. This symbol by the alpha time, alpha angle. if and in later slide I'll explain step by step what is the R, what is K, and what is the alpha. And the next, maximum amplitude. Maximum amplitude basically represents the interaction of platelet with the clotting cascades. And finally, after the clot will form, and then last step is the lysis. So lysis of the clot is by the steps called the fibrinolysis. So this thromboelastography instrument is basically assay system which assess the measure the components of thrombus formation. So there are 4 important components required for the thrombus formation. As you can see on the slide, first one is the platelet aggregation, clot strengthening, fibrin cross-linking, and fourth one is the fibrinolysis. So what is the platelet aggregation? So once platelet is activated, these activated platelets interact with the another platelets and release their content to release their content to show the autocrine and paracrine manner and bind with more platelets. So in this manner, these platelets getting aggregates. Next step is clot strengthening. So for to heal a injury, only platelet clot is not the much stronger clot. So to reinforce the clot strengthening, we need the fibrin. So fibrin synthesis are clotting factor which reinforce the platelet aggregates of platelet, make it stronger. And next one is the fibrin cross-linking. So now when clotting cascade is active, there is formation of fibrin and fibrin polymerization, which behave like a glue structure, and this glue structure help in the more strong more strengthen this clot of aggregates. And finally, this aggregates is getting fibrinolized. So here in this slide we are showing how TEG perform these assays and how this recorded the thromboelastogram. So you can see in the red square box, and we are zoom out in the right side corner. So you can see there is a cup, as a name mentioned, the cup, and this cup is full with the blood. We've been decline
करो उसको.
So this cup is full with the blood, and this is volume of 0.36 ml or 360 microliter of whole blood, and this blood is taken from the kaolin vial. So this is the kaolin vial and citrated kaolin vial, and this blood, this cup is surrounded by a heating element which maintain the temperature 37 degrees centigrade. And in this blood, you can see there is the inserted a pin which are dipped in this blood, and upper one is the torsion wire which are finally connected with the electrochemical transducer. And this cup and pin rotate 4 degree to 45 minute angle, and this rotation was recorded by the electrochemical transducer. So once this rotation is start, and clotting is getting clot formation is started. So clot formation is started, the movement, the rotation of this cup and pin is hindered, and this hindrance was recorded by the electrochemical transducer, which are finally exported in the graph that are called thromboelastogram. So there are 5 different parameters which we can interpret from the thromboelastograph. So these 5 parameters are reaction time, which takes 4 to 8 minutes. This is the normal range. K time, this is the clotting time. This will take 1 to clot kinetics. Key time is the clot kinetics. This will take 1 to 4 minutes. And next is the alpha angle, which is normal range is 47 to 74 degree, and maximum amplitude. This will range 55 to 73 millimeter of clot that will form in that cup. And finally, lysis of that clot, which is directly proportional to the fibrinolysis, and this normal value is 0.28%. So a step-by-step description of all these parameters explained in the next slide. So first we'll explain about the reaction time. So as you can see in the graph, left side you are seeing the normal, and their R time is very short, and this is the normal range for the R time. And in next right side graph you can see there is the prolonged R time. So this will represent there is delay in the clotting cascade, mechanism. So R time is the time taken by fibrin clot to form initially, and it depends on the clot factor. Suppose this clotting factor is any patient which has the deficient in the clotting factor. So we find a graph which are present in the right side, and what treatment clinician is follow. It is by the FFP, fresh frozen plasma, to stimulate the clotting cascades. Next is the K time, so also called clot kinetics. So K time is the time taken by the clot to reach a fixed strength of 20 millimeter. So once 20 millimeter of a clot strength is reached, then that particular time to have the 20 millimeter of clot is called K time. This time is also depend on the clotting factor. So there is a case if K time is prolonged when blood has low fibrinogen. So is there, if any patient which has the low fibrinogen, then we have found an K is getting prolonged. So clinician follow the what treatment as given by the clinician. It is the cryoprecipitate. Sometimes it is if cryoprecipitate is not present, then it is FFP, frozen plasma, is also given to treat this situation. Next is the alpha angle. So what are the alpha angle? Alpha angle, it's mean ki it reflects the speed of fibrin accumulation and polymerization. So once fibrinogen is getting converted into the fibrin by the clotting cascade mechanism. So how this fibrin is getting accumulated with that clots that are called alpha angles. So alpha angle is the slope of take pressing drawn between the R and K values. So this alpha angle is low. This will also reflect the low fibrinogen, and the clinician follows the treatment cryoprecipitate for the low alpha angle. In the next slide, I'm going to explain about the maximum amplitude, which are the next parameter of take interpretation. So maximum amplitude is the highest vertical amplitude of the take tracing, which measures the overall stability of clot. It means maximum amplitude you can see in the graph. Here is the normal graph, and in the right side there is the abnormal graph which shows the reduced maximum amplitude, and this maximum amplitude is depend on both platelet as well as clotting cascade mechanism like fibrin. So if there is if there is the interaction in both platelets and fibrin, then we have a normal maximum amplitude. And you can see in the right graph there is the reduced graph, and this will show there is deficient in platelet number. So clinicians follow the transfusion of platelets. So this cases was seen in case of thrombocytopenic patient, and abnormal platelet function will decrease MA, maximum amplitudes. And next is the lysis LY30. So what is the LY30? LY30 measures the rate of amplitude reduction. So you can see in the previous, this is the maximum amplitude, and there is the maximum highest height of this two upper and lower graph, and you can see, and this graph is the maximum, and the rate of decreasing this slope, the rate of decreasing slope is called lysis. So lysis, LY measures the rate of amplitude reduction 30 minutes after the maximum amplitude is reached. So once maximum amplitude is reached, then after the 30 minute lysis will start. So and the range of LY30, as I have mentioned earlier, 0 to 8% is normal. If it is reduced, then there is a problem in the patient, which shows the fibrinosis increase the value of LY30, and the treatment which are given to the patient are antifibrinolytic agents. So in conclusion, I just mention all the 5 parameters in a single slide. So you can see here that I mentioned here the TEG and the ROTEM. So ROTEM is also another instrument like TEG. So which there are two different terminology they are using. So in the case of TEG, we are using reaction time or R value, while in case of clotting time in case of ROTEM, and this is the CT. So what is it? It is the time till initiation of fibrin clot formation, and what are the normal value? It is 5 to 10 minutes. Sometimes it is 5 to 8 minutes. And abnormality is what? R value is getting high, and if R value is high, then problem is the clotting factors. And what are the treatment? It is given by FFP or the protamine. Next is the K value. K value, or in case of ROTEM, this will termed use clot formation time, that is the CFT. So what is it? Time to achieve 20 millimeter clot on assay representing thrombin platelet interaction. So finally this will stimulate the fibrin polymerization. So now what are the normal range of this? It is 1 to 5 minutes, and what are the abnormality? K is getting high, and this will represent the low fibrinogen in that particular patient or that particular person. And what are the treatment that are given to this patient? It is the cryoprecipitate or fibrinogen. Next is the alpha angle. Alpha angle, what is used in both TEG as well as in the ROTEM. And alpha angle, as described, rate at which fibrin cross linking occurs, or you can say how fast fibrin is getting accumulated with that clot. So what are the angles? 45 degree and 45 degree to 75 degree. So if alpha angle is low, then fibrinogen is also this will reflect that there is low concentration of fibrinogen in that particular patient blood, and this will treated by the cryoprecipitate and fibrinogen treatment. Next is the maximum amplitude, or in case of ROTEM, it termed as maximum clot firmness, and MCF in case of TEG, it is the MA. So this is the maximum strength of clot, and what are the normal value? It is 50 to 75 millimeter. So maximum amplitude can be high, can be low. If it can higher, then this will reflect the hypercoagulable property, like which can cause the stroke and other. And if it is low, then this will reflect that there is low concentration of platelet in that particular person or patient. So what are the treatment? Clinician follow, it is the platelet transfusion or DDAVP treatments. Last one is the LY30. It is the lysis after 30 minute after maximum amplitude, or in case of ROTEM, it was named clot lysis, represented by the CL. And what are the normal age? It is the 0 to 10% or 0 to 8% sometimes. So if LY is higher or CL is higher, this will represent the clot breakdown is faster, or fibrinolysis is faster. So for this treatment, clinician give the patient to the anti-lytic agent to treat the situation. So as you can see in this slide, here we are showing the normal take tracing which are captured in all lab. So this is the graph this is the sample from the Kevlin vial, and you can see there are the numerous parameters. First one is the R minute, O to 8 minute, K time, and alpha angle, maximum amplitude, and G and EPL, AMM and CI and LY30. So we are basically track the R, K, angle, maximum amplitude, sometime EPL and LY30. So you can see here in this graph, this is the normal tracing. In the next slide, I'm showing what are the abnormal tracing for the tick. So you can see here the two situations that we're taking. First one is the hypercoagulable state, and next one is the hypocoagulable state. So by look at this graph, you can just simply find out the difference. So in first hypercoagulation, you just see the R time. So R time is very small, and in case I'm hypocoagulable, this R time is larger. So this will represent there in case of hypocoagulable deficient in the clotting factor, and in case of hypercoagulable there is hyperactivation of the clotting cascades. So if R time is high, then K time is low, and alpha angle is high. Alpha angle is high, maximum amplitude is high. LY time is also high. So this situation was seen in medical cases, sometimes it is in COVID patient, deep vein thrombosis, ischemic stroke, heart failure patient, and pulmonary embolism cases. And this situation, to assess to know the situation of that particular patient, a clinician check the trauma condition and liver transplantation. Is it blood is hypercoagulable or hypocoagulable or in normal state? So hypocoagulable cases we have to see in the dengue, malaria, hemorrhagic stroke, and other. So you can see this is the paper. This is the published article in International Journal of Nanomedicine, and it is titled "Effects of Nanosyria on Human Platelet Function and Blood Coagulation." And in this publication, here we are using the thromboelastograph to see the effect of nanosyria. Nanosyria is the cerium oxide which which is as a pollutant which present in our surround environment. So how this nanosyria will affect to own blood or own circulation, own platelets. So you can see, so with the increasing dose of nanosyria particle, cerium oxide nanoparticle, we can see there is decrease in the R time, and we can see there is the increase in alpha angle. So this will represent the higher dose of this nanosyria will create the hypercoagulable state for that particular situation. Another publication from this is the recent publication published in the eLife, titled "Notch Signaling Function in the Non-Canonical Juxtacrine Manner in Platelet to Amplify Thrombogenicity." In this publication, figure number 6, we are using the thromboelastography, and you can see one is the control, another one is the treated. So green one is the treated and black is the control. So green is the drug, it is the DAPT drug that will increase the R, that is the reaction time and clotting, and we can also see this will decrease the platelet activation pathway. Suppose a platelet is getting hypercoagulated, so this this DAPT is down regulate the platelet activity and inhibit their thrombotic behavior and procoagulable behavior. So this instrument I'm just showing with the very in zoom form. So you can see on the top there is the yellow color key that are the motor key which maintain the temperature of surrounding of the cup at 37 degrees centigrade, and as well you want more or less. And green color button is the power button which if we press then and instrument is ready to perform an experiments. So here in the second lower one is the, you can see there is the rod, and that rod is held with the carrier, and this in this carrier you see the well where we put the cup and pin, and this cup and pin in which we are add the calcium chloride and whole blood sample taken from the kaolin vial, and this finally placed up to the black vial, and you just and fix with the and this all the procedure I am going to show you in the next video as in demonstration video. So thank you for the listening me, and ask any your queries regarding this. Unmute all. Unmute all.
एक काम करो, पीपीटी पर एंड कर देते हैं तो मेन स्क्रीन आ जाएगा.
Yes. Stop sharing. Yes.
हाँ, ठीक है, म्यूट हो गया है.
Yes.
Hello.
Hello. Is there any question regarding this lecture? Please ask.
चैट बॉक्स में भी देख लीजिए. नहीं है कोई क्वेश्चन तो फिर आगे वीडियो क्वेश्चन.
अभी इनका all are mute. Unmute na? Is there any question regarding this session? Please ask. Am I audible? Nature of sample use. ये actually this will अच्छा okay ठीक है. The nature of sample use is only whole blood, or we can use PRP or was sample. So one question from the Pooja Yadav. So nature of sample use in only blood whole blood, or can we use in PRP or was platelet? So in was platelet we cannot use the TEG, but for sometime we have we in few publication we have seen some lab use the PRP in for the TEG analysis. So most of the time whole blood is used in this technique, not PRP and was platelets. Is there any more question?
No. Okay. If there is no question further, then we can proceed with the video part, virtual lab.
Yes. No question, sir.
Okay.
So play the videos for the.
Please play the video.
Oh, okay. ठीक है. Yes. And वॉइस इज म्यूट, यस ठीक है, म्यूटेड रहेगा, ठीक है।
सबको म्यूट कर दिया ना, म्यूट ऑन कर दिया।
ऑल आर म्यूटेड ना?
आवाज आ रही।
म्यूट ऑल कर दो।
म्यूट ऑल।
पता नहीं रहता कि उनका माइक ऑन है।
So here in this video you can see there is an instrument called which are in our lab. This is the thrombolastography, and here are two key that we will explain in the our lecture. So first we press on the green button.
See here, there are two panels, chamber one, chamber two. You know the chamber when the same someone, there is a two rod like structure called white color platform. This platform today also oscillate when you go to 45 degrees in this panel. This will rotate the sides. Similar to this chamber, the formula also rotated 45 degrees. So here is an when we place the cube above, see the
म्यूट कर दो।
So you can see here this is the cup that we have taken in our hand, and upper one is the pin. This is the pin and this is the cup, and this cup and pin was placed in the cuvette well switched in the carrier. So after the placing we take this carrier up and press very strongly. If there is any gap, small gap in the black and white, so we have an error in our data. So we just press it very tightly. And to fix the pin, there is a lower side in a small push button. We just push it for 3 times, 1, 2, and 3. So 3 times push, and then we'll fix this. So pin is up in the black folder, and cup is there in the white carrier. So you can see this is the kaolin vial. In this kaolin vial, we will take the fresh human blood sample drawn 1 ml, and after taking 1 ml, very slowly we are mixing it 1 to 2 times, and this is the fast process, because if we place it for the longer time, this platelet is getting activated in the vial. So we'll just mix it for 1 to 2 times and very slowly, and take first we add the 20 microliter of calcium chloride in the cup, and after the placing this calcium chloride, we just take 360 microliter of kaolin blood and add into this vial cup.
So first we have to add the 20 microliter of the 0.2 molar calcium chloride. So after the calcium, we have to add 360 microgram of the blood taken from the captive one.
This addition does not. This mixing of this addition was not need any mixing of blood and calcium chloride. We just simply add the blood and take it up.
And you can see here in the folder, there are three places: descent position, load position, and base position. So here using load position, and if we click this load the base position, then somebody is ready to for an experiment. So, so first.
So before we set it to the test position, we just first we open the software, and software here is in the green color named TAG, and this software open needs some security option.
The password. Now, on the tape, let's see the tape. And once we click over the tape, then you can see when you see the dialog box, you can see the passing the values and I'm clicking the label.
So while we are starting the experiment and open the software, we have in a dialog box, and this is the calibration dialog box where we can first we have to e-test for the channel A and channel B, and both channel are in equilibrium, and once both channel are in equilibrium, then the instrument allowed to perform our experiment.
After this, we first we have to remove white black is no important, and then we start the e-test. First we start the e-test. Here is the one channel one. And then this should be made. This should be less than 10 both channel have the differences. Than the now set the channel one. For the two, and that's the sample. See the frequency control and then channel. You can see what design for the algorithms we are going to scientific delivery. You can see the date. Because we are using signal then to start this cycle. You can see here there is the three color, active color is selected. So you can see this is the active and along the two and color. After that we have this is see here. See here, we have to R here. R then from start and then start studies. Yeah. Sample
So you can see this is our lab graph, and this is not done at all the time. So you can just see here the R time and K time, angle, maximum amplitude. Here is the APL and the lysis 30, and which will shows in the earlier lecture graph. This is the normal graph, and this is the control sample. So after this we can over and open for question section. So you can ask your queries. इसको बंद कर and unmute all. Okay. So you can ask your queries regarding this lecture and videos which I have played regarding the thromboelastography.
Good morning. I wanted to ask you in which vacuum cleaner does the blood sample received from the wards or from the patients in the lab?
I have collected sample in the acid citrate dextrose vial and our citrated vial. Let's see. Yes.
Fine. Thank you so much.
Yes, ma'am.
Any more questions?
Had any bubble during the loading? Okay. Okay. Yes. Any more question? No, then it is over.
Hello, this is Iban Lahadem from A.M.S. Kalyani.
Yes.
Actually, I'm very new to all this technique. So, what is the importance of this? You having this instrument? Is it like to start a platelet biology lab or something like that? Is it like one of the initial instruments that somebody has to have it or something?
Yes. Can you please repeat your question? I'm not getting you.
Hello, can you hear me?
Yes. Yes.
No, actually, see, I'm very new to this. So what I want to know is what is like the significance of this instrument? Is it like very important to have this instrument to start your, if suppose I want to start something on platelet biology, is it like one of like the top instrument that has to be there in the lab or something?
No, this is not like in this very much significant. If you have, then this what is the beauty of this instrument? This instrument assess the different clotting mechanism in a single panel, like how clotting cascade is getting activated and how the platelet is getting ready. So there are different parameter R time, K time, alpha angle, and maximum amplitude and lysis. So reaction time of clot formation, reaction time of clotting cascade activation, and alpha angle, how fibrin is getting accumulated, and maximum amplitude is the interaction of platelet and clotting cascade, and lysis is the fibrinolysis. So all these parameter in the single instrument we can assess. So and this is not very much necessary. You can use, if you do not have, then you can another assess also.
And also, if may I add, this is a good instrument for a clinical collaborative part. Like if you have a cardiology unit who are doing fibrinolysis, or if they are having many of the clotting, so this you can also use for clinical reporting as well. So if it suits your demand, definitely you can have.
So what exactly like the price of this instrument? Any?
Around 15 lakhs.
15 lakhs and more, ma'am. Because most of the instruments are old to our lab, so definitely we will. If you email us, we will share you the contact details of the person, company representative, and then you can negotiate. Okay.
Center facility.
Yeah. Hello.
Okay, thank you. Yes.
Okay.
Thank you.
Does it make any difference if you use the anticoagulant apart from? Next question is from the Pooja Yadav to everyone. So does it make any difference if we are use anticoagulant apart from the citrate dextrose? So you can use another anticoagulant also, but for the particular for a particular anticoagulant instrument, our company will provide the different for the kaolin vial. For the kaolin vial, we are using citrated, And for the another anticoagulant, this company provide the another named vial to dilute the sample. So I don't know the what are the vials name. I'll
So
mail you.
basically, as per your different type of blood storage, the vials need to be shuffled. So you need to go for the company instructions for selection of the vial. Any other questions?
I have another question, which is not related to this,
Okay.
but basically, I mean, platelet has to be stored within the 22 to 25 degrees, right? So do you have like an incubator or something, or is it like you make sure that the room is like at that temperature or
Uh,
something like that?
So
Basically, the laboratory setup is being set with the air conditioning. And if you want to store the blood, then we use generally the blood banking facility.
Okay, okay, thank you.
One question from the Vishnu Priya: can we use sodium citrate solution as the anticoagulant? So we can use the sodium citrate while blood also for the kaolin oil.
So next is the small deliberation on biostatistics for health professionals or say biological students. It will be given by Dr. Barul Sharma from Department of Physiology. He is also enrolled as a PhD student in our department, and he will have a small deliberation on this particular topic. So I welcome Dr. Barul Sharma. Please be.
मुझे इंट्रोड्यूस करना है।
नहीं नहीं करने की जरूरत नहीं है, अरे मैं खुद कर दूंगा।
कहाँ है?
मेरे मोबाइल लेना है, मोबाइल मोबाइल करना चाहिए।
And also sports medicine. Okay. So he has done his sports medicine degree from NIS Patiala. He has also worked in A.I.M.S, New Delhi. And his area of interest is sports medicine, clinical physiology, and exercise science. And he's very good in applying so many statistical tests. So we have invited him for a small talk. He'll show you. And here, okay. Sorry for the delay. He has received so many awards regarding his research in physiology, and he is तुम्हारी सोसाइटी का नाम कहाँ लिखा है?
इंडियन सोसाइटी ऑफ स्पोर्ट्स एंड एक्सरसाइज मेडिसिन बोल दो।
कहाँ लिखा है?
ना current president
Yes.
तो number 5
He is current president of Indian Society of Sports and Exercise Medicine. And its areas of sorry for that.
Mute yourself, you cannot hear me.
Can you hear me? हो गया? Hello? अच्छा, okay. ठीक है, then let me share the screen. So thank you very much for giving this opportunity. I am Dr. Hanjaban Barun Sharma, and I am medical and clinical physiologist as well as sports and exercise medicine specialist. And right now, since my interest is in clinical and interventional, metabolic biochemistry, so I have come here to learn the biochemical technique here. So biostatics, I have been in the applied aspect of biostatics since I was working in the Sports Authority of India, deals with different national players. So let's begin a talk, and I will be speaking only on the applied part because of the time constraint. Previously, everyone, we basically conducted a workshop under the same banner in 2021. With collaboration with the names. So in the future also we will be planning to make more detail on the workshop, but for now I will be talking only on the basic because of the time resting. So not going into detail of all these things, what is important, I want to highlight this, what is the importance for the knowledge of biostatistical technique. It's for the better study plan and design. Before just doing the study, also your knowledge of the basic biostatistics will help you in planning in a better way, as well as data analysis and interpretation and application of that knowledge into more advanced, applied translational research. And remind you, there are lots of research published paper which has lots of statistical error. It may be related with study design or methodology, data analysis, data presentation and all of these things, and based upon that different what you call a paper has also been published. That means statistical error is there even if you see in the good journal. So that's why this paper is important. It was published in the British Journal of Sports Medicine, and they have highlighted the list of the different checkpoint where you can see before going into the application. But now let's go into the what you call what is relevant to this talk. That is, statistics teaching is very simple. It is just straightforward. But the problem comes when you apply it. That's why application of biostatistics is more important. And with the presence of lots of software like R, Stata, SPSS, even Excel also, you can do lots of data analysis. So what I want to focus here is the one thing, that is number one. The most important point of beginning in biostatistics is always you have to check your sample size, which may be relevant to your research or not, but I will not go into the detail of it. And another one is a normality testing. Most of the time, normality testing is not done, which is wrong, because normality testing and testing of the assumption of different statistical tests, which you will be applied, you have to do it before going. Then outlier, because the outlier, every data interpretation, your result, your p-value, everything will be influenced by your outlier. And missing value treatment is also very important. And another one, apart from the normality and the sample size, one has to understand the homogeneity of the variance, which is also known as homoscedasticity. You have to know how to test. Why I am focusing only on these 3 points, because these are not done at all. People just apply, parametric impurity tests and without doing any kind of statistical requirement. So this is basically wrong. That's why this sample size calculation, I will not go into the detail. There are different formula. G power software, free software is there. So I will not go, but I want to say about something about normality testing. So in normality testing, it may be skewness may be there, or it may be kurtosis, and all the software, even in Excel also, it will give a numerical value for say kurtosis, and that kurtosis, that value should be approximately equal to 3. Then only you can say that it is nearly, normally kurtotic. Otherwise, it may be platykurtic or leptokurtic. And regarding the skewness also, so directly going into my this thing, because lack of time is there. So there are different formula are there if you want to go into normality testing. Visual method is everyone know, histogram everyone know, box plot, box whisker plot is important. PPQ, QQ plot, and everything is there. Then 1 to 3 tests I want to say there is Safirovic test and a Kolmogorov-Smirnov test or D'Agostino-Skewness test is there, which you have to apply before any kind of the data analysis. Without that assumption, and if assumption and limitation is not satisfied, then you cannot go ahead. Your p-value is not reliable at all. So that's which you have to understand. Just like kurtosis, also one value for skewness is also there, and that skewness value has to be between minus 1 by half to plus half. Then only you can say that your data is approximately normally distributed. So how about outlier detection? Because outlier, your mean value is highly influenced by outlier. You cannot rely on the data. That's why the outlier treatment or outlier this thing has to be done. And the value which we follow is traditionally 1.5. If are using SPSS also, you can see that if you use a box whisker plot, this is the box whisker plot, and if some value is given by a circular point, that means that value is an outlier that has been described by your software, that is SPSS. Traditionally, what we use is any value which falls more than 1.5 times your interquartile range, that is IQR. IQR is interquartile range. You can calculate it by using your 75, your 75 minus your 25 value. That is 75 is also known as Q3, and this is Q1. And in that way, you can see whether a particular value is lying outside that zone. If it is lying outside the zone, better remove it. But the problem of outlier removal is if you remove too much outlier, then what will happen? Your data sample size will decrease, and this will affect your the power of the study. That is 1 minus beta. That's why you have to understand how much outlier to remove, and hence this traditional formula 1.5 we don't use nowadays. We accepted is 2.2, and SPSS if you are using SPSS gives the value of 3. If this comes a star mark is there, then it means it is an extreme outlier. You should remove it, because whole your entire theory will be challenged by the presence or absence of outlier. This one has to understand. And missing treatment is also important. Sometimes if you are using pre and post data, then sometimes because of some mistake or because of your loss in follow-up, your post value may be missing. And if missing value is there, then whole analysis will be affected. So how we are going to deal with, like for example, another is known as ITT, that is intention to treatment, where you have to replace that missing value by just p-value. Or you can use there are different statistical method like mean and all this thing you can use to correct the outlier. One point which I want to say is in case of the SPSS or any kind of software like Stata, like R, you have to say how you are going to exclude that data. There are two ways, that is known as the pair-wise or list-wise. In pair-wise, if you select that pair-wise or case-to-case wise, then exclusion of that particular value from the analysis will be done. That is missing value, but whole case will be there. But if you do list-wise, then what will happen? Entire case will be removed. So this thing one has to understand before going ahead with the data analysis, which I will be showing. So I will not go into the detail of this thing. One point which one has to understand is too much focus on the p-value 0.05. If it is more than 0.051 or 0.0449, then problem is there. So p-value, don't focus too much on p-value. P-value is what you said, that alpha there is what you said, 0.05. What is more important, which you have to understand, is always and always focus on confidence interval. If you are giving some data, then you have to report a confidence interval that is 95 confidence interval. This is very important. And number two, you have to focus on the effect size. How much is the actual difference? That quantity, this is more important as compared to the p-value. P-value doesn't give the quantification of how much important is your intervention. You have to remember, because people just think that p-value is nothing but a probability of the alternative hypothesis. Is wrong. This concept is wrong. P-value is actually, instead of the p-value, what you are doing is you are assuming the null hypothesis. That means if you are doing some kind of the intervention, and if you are doing pre and post, flow cytometry in case of the platelet by adding something, then you are assuming in null hypothesis that there is no effect. So p-value, for the interpretation of p-value, you have to assume that there is no effect, and that p-value, whatever effect you are getting, is purely because of the random error. That's all. So p-value, if the more p-value is there, that means what your random effect is not. That means your treatment effect is not there. So that's why we require a p-value which is very, very less. And before going into the research detail, we set the alpha 0.05. That's why p-value is not so important as as always has been focused. More important is effect size, and more important is confidence interval. So this one has to understand. Different statistical method are there depending upon whether your parameter is your data is parametric or not. So I will not go into the detail. There are different flowchart is there. What I want to show is your data has to be both reliable as well as validity. For that I will just show how I have to process forward with some detail here. So let me share one slide regarding SPSS. I will just show in SPSS whatever I talk. Then after that, I will show in Excel. Those who are not familiar with the SPSS, you can simply do in Excel also. So let me show the data.
Share screen.
Okay, share screen. कहाँ से होता है? Okay.
Share your screen.
So can you share my data? So let me just, these are a sample from 1 to 15. This is just these are the three data. Let's focus on this first three data, 1, 2, 3, and this column is also there. So basically what is happening is, remember the last day, previous day, Dr. Josna was speaking some platelet intervention. This is just data of that. So I just extrapolated in and let's apply some biostatistics and see how the result goes. So here it is a two group. This is for mitotract signal with no treatment, and this is the added nanocedia. So if you want to compare between this and this, you can compare by peer t-test because this is a peer sample if normality is satisfied. Otherwise, you can do unpaired t-test also depending upon your research question. So let's apply the paired t-test and unpaired, the non-parametric version also. So before going ahead, what we have to do is we have to do the assumption for normality test. So let's apply the testing in this column. So for that, in the SPSS, users have to go to analyze, to the descriptive statistics, go to the explore, and in the explore you can just check what this is the treatment and no treatment with the nanoceria. Just import this data, then you can go to the statistics part. If you want to see the outlier, you just click here in the outlier. And if you want to see what are the things which you require, like for example histogram, I required histogram because I want to see whether it is normally distributed or not. You can see the different plot like a statistic, like a Safirovic test. So you just click here, and after that, and there is, so remember what the exclusion. If there is missing value, then you have to do list-wise or case-wise, exclusion. That's why this is given, because your p-value and the numerical value will different depending upon whether you are excluding list-wise or pair-wise. Let's check pair-wise in my case, and let's see and what the SPSS has to say. So this is the SPSS output. So this is given. So you can see that, just see there are different values are there. For example, 95% confidence interval is there. Sometimes what is happening is if you are dealing with a non-parametric data, non-normally, and if you are using the median, then 95% confidence interval for median is different from mean. So people, usually report the 95% for mean, which is wrong. So if you are using median, then you have to use 95% confidence interval for median. So it doesn't give you that thing. You have to manually calculate and make some algorithm in the SPSS. Then only it will give. See, the skewness is also there. Remember, the skewness has to be between -1/2 to +1/2, like that. Process, approximately 3 and all this thing. So this value is there. By visually looking, you can see whether a particular data is normally distributed or not. Otherwise, you can always go to the test. So there are two tests here. You can see that. Remember, I have given to the Safirovic test and Kolmogorov-Smirnov test. Which one to use? It's always better to use both. But if you want to rely only on one test, then it's always better to go with Safirovic test, which has more power. But the problem with Safirovic test is your sample size is very, very less. Say less than 10, less than 6, less than 7, then it is not reliable. So that's why you have to use this test. And what you want to see is the p-value. The null hypothesis of this Safirovic test or any kind of the normality testing says that your data is normally distributed. That is the null hypothesis. So if you want null hypothesis to be true, then what will happen to your p-value? Your p-value has to be high. That means you want this p-value to be more than 0.05. Then only you can say that the particular data is normally distributed. So here in this case, my two data are normally distributed because the p-value is more than 0.05. That means I can apply the unpaired t-test here or paired t-test here. So these are the different, the algorithm histogram. It is not entirely just like a bell-shaped curve, but still some, something is there. You can find out approximately equal to normally distributed. And this is the leaf and the stem plot. And this is the QQ plot where, all the dots line has to line along in this line, then only you can say that this is normally distributed. So, and this is the box whisker plot, and in this box whisker plot, you can see that it is approximately symmetrical. That means it is slightly normally distributed. So in this way, you can always see the finding. So after knowing this thing, let's go and do the testing, that is unpaired t-test and all this thing. For that, what we can use for comparison, just let's try to see the paired t-test. That is after adding of nanoseria, what is happening in that mito tracker value. So paired t-test, just select it, and you want one variable. Your variable is no treatment, and another is the added nanoseria. Just add it here and see, check the variable. Again, it's again said that which one to you want to select by exclusion, by analysis for analysis, or list wise. That means if there is any what you call missing value, that's why missing value addressing is very important. People doesn't know that, they just do it like that, but it is wrong. So by default, case by case analysis selected is okay, and then you can go ahead and click here, and then you can see whether your this thing is statistical significant or not. Like for example, here you can see that it is statistical significant. That means by adding of this nanoseria, your data is statistical and p-value is 0.004. Remember, p-value doesn't tell you about the strength of your the finding. For that, you have to use what? Effect size. And most of the time, for like for example, effect size, you can use for unpaired t-test, you can use Cohen's d. Cohen's d value you can remove. And another one, if you want to, suppose if you want to do unpaired t-test, then how you are going to go ahead? This is for paired t-test I am showing. The same data I have arranged in a column wise. Like for example, same data I have arranged here in this way. So from here to here I have arranged, and I have coded the value. Like for example, here in this coding is then one means no treatment added, and then second means addition of the nanoseria. So I have arranged that data in a column wise to in a single column, single file. So why this is important? Because this is important if you want to analyze by using unpaired t-test or student unpaired t-test in SPSS. Excel doesn't require all these things. For SPSS, you have to do certain modification. So let's see how to go about and doing what you call the unpaired t-test in SPSS. So let me check. ये क्या आ रहा है? So let me check the unpaired t-test. Just select the independent sample t-test. Then you can select, and it says the grouping variable. That means grouping variable with the code which we have used. Remember, I have used one for no treatment, two for added nanoseria. So remember which one to use. I have this one. That is, it is a what type of data it is? It is a nominal data, categorical data. So just put here categorical data and define the group. That is 1 and 2. I have just defined 1 and 2. And whatever you want to put here, just put there. For example, this is the mitotracheal data, this thing I have put here, and you just see whether, similarly, if some missing value is there, then you have to choose which one. By default, it is always better to choose analysis by analysis or tier-wise exclusion, which is by default also selected. So let's go ahead and see. This is the independent t-test. So in independent t-test, you will get all the values you want. Remember, standard error of mean. You should not use standard error of mean in case of the analysis. People use standard of error means like anything. Why? Because standard of error means is formula by standard deviation divided by root over n. That is sample size. So standard error is always smaller as compared to the standard deviation. And because in order to show that your variability of the data is less, that's why the author usually represent mean plus minus standard error, which is wrong. Standard error is actually saying the variability of your population mean as compared to the your sample mean as compared to the population mean. So that is have different value. So standard error of mean don't report. Many papers have been published saying that the reporting of standard error in good reputed journal, they are wrong. And so always use your standard deviation if it is normally the other. If your data is not normally distributed, then use median plus minus interquartile range. or quartile deviation. Quartile deviation is interquartile variance divided by 2. That thing you can use. So let's check here. This is the output in the SPSS. And remember, I have said that assumption of normality is important. Apart from that, there is one assumption which you have to satisfy. That is the homoscedasticity, or assumption for homogeneity of the variance, which people doesn't do at all, but that is wrong. So in SPSS, Levin's test is given automatically, where you have to check the p-value. Here the p-value is 0.23. That means it is satisfying your homogeneity of the variance. That means your variance is approximately equal. SPSS has given two p-value, so don't which one to use. It depends upon your p-value of the Levin's test. If the Levin's test p-value is more than 0.05, that means you are satisfying the condition of equality of the variance, and hence you will use the p-value of this row, that is 0.240, not 0.241. Many a times these two p-value are different. That's why the certain basic statistical knowledge is very important. So this is for unpaired t-test. So let me go ahead with the how, if suppose if your data comes as an unpaired, what you call non-parametric, how to go about in the testing of non-parametric. Similarly, you can just click here non-parametric test, and in what you call the independent sample, or in the legacy dialog, you can go ahead and suppose I want to check the non-parametric test of the unpaired t-test. That is, just select here, that is 2 independent sample, and similarly everything is same. Method taker, the code I will put here, and I will just mark 1 and 2, because this is the code which I have used, and the signal I will put here, and I will select Mann-Whitney-Yoot test, because Mann-Whitney-Yoot test is a by default the non-parametric version of the unpaired t-test. What is a non-parametric version of the paired test? Wilcoxon sign rank test. What is a non-parametric measure or repeated measure ANOVA? It is the Friedman test. So one has to understand all these things basic. And let's go ahead with the, if you want descriptive, if you want quartile deviation, you can, again, their SPSS is asking of how to treat the missing value, because missing value treatment is very important. You cannot neglect that. So I will just go and see the mean value, and this is the p-value which is given for what you call the Wilcoxon sign rank test, and it is not statistical significant. In this way, you can calculate the comparison of two groups. Suppose you have a 3 groups. So let me give some basic of how to analyze 3 groups. That is one way ANOVA and all these things. So this is the this is another file. Let's check this file. This is, for example, this column. Just see this column, this one, and this one. And this column is for the effect on the fibrinogen binding, that is MFI value, with addition or non-addition of nanoseria. Suppose this is the no addition of the nanoseria. This is the addition of the nanoseria, say a 50 dose. Then this is the 300 dose. So if you want to compare what is the effect of the increasing dose of nanoseria on say fibrinogen binding, then you can use the one-way ANOVA, or if you are using repeatedly, again, that it becomes in a repeated measure ANOVA. So if I suppose if this 3 satisfy the normality, so how you are going to go ahead with a repeated measure ANOVA? So here similarly same thing, you just click here in the repeated measure ANOVA. Since repeated measure ANOVA comes under general linear model, so in general linear model this repeated measure is given. So you have to know where that particular statistical test is there. So just click here, and it will ask factor. How many factors are there? 3 time point is there. Because 3 you are measuring a sample in 3 way, so you can put here. You can use one way ANOVA also. So here I have put 3, and I will just add, and I have to define what are the 3. So the factor, let me check. Okay. Sorry, factor name. So sorry, wait, let me check here. So this is the repeated measure ANOVA. Similarly, same thing, factor 3 levels are there. I will just and I will define what are the factor. So define one factor is no treatment. You just put here, and you just click here. It becomes your group 1 is your no treatment. This is the 50 dose nanoseria. You just put here, and this is a 300 dose. You just put here. So everything is there. And if you want post-hoc analysis also, you can do. Like, for example, bone foreign correction is very important. Correction of p-value is very important if you do multiple comparison. Otherwise, your 0.05 is not reliable at all. So this thing one has to understand. So just by putting here, if it becomes significant, then only you can do. And these are the option. Similarly, what you want, like for example, homogeneity of the variant testing is very important. Yeah, you can see comparison. You can do also like bone Veroni correction. You can do, and if you want different, effect size also you can select here. So the estimation of effect size is there. So everything is given in SPSS. You just go ahead and see what is the effect here, and you can see this is the table which is given for what you call the repeated measure ANOVA. So, in case of the repeated measure ANOVA, p-value is there. Similarly, just like Levin's test was used for testing of the what you call homogeneity of the variance. In the same way, in this year, we use the Mousyl's test of the homogeneity of the variance. And you have to see before going into the p-value. And that p-value is given here. It is significant. That means it is violated. We cannot go ahead and simply take the p-value of the repeated measure ANOVA, because your assumption of homogeneity is violence. Then what will you do? You have to go into the correction. That is the most of the time which we generally use the Greenhouse-Geiser correction of the p-value. So you have to select Greenhouse-Geiser p-value here. That is 0.234. Many a times they are same, but many a times also it is different. So you have to understand which one to go ahead and which one to not go ahead. So all this thing is important. This is what I want to say. And many a times people also how to make graph also in SPSS. Everyone must be knowing how to make graph. Like for example, if I want to make a graph by a bar graph comparison between pre and post two factor, then I can use the graph. Like suppose select here, go to the legacy dialog and scatter. In scattered plot also you can make. Or chart builder, you can just select the chart builder. You just drag whatever you want here. And if you want two group comparison, then remember my two group was this one. It is the mitotrack signal. So mitotrack signal just put here in the y-axis. And in the x-axis, what do you want? You want two group. And this two group you just put here. And so this become no treatment and added nanoseria. What is the graph will be there. And if you want to add an error bar also, you can always add error, like for example, display error bar. Error bar, it can be done mostly on the standard deviation, one standard deviation we generally put. So users put one standard deviation applied, and let's see, this is what, and this is the graph which you can get in SPSS regarding your this thing comparison with the error bar. And showing error bar is very important. Otherwise, people doesn't know what is the variability of your data. So error bar must be given. And many times people have said, I don't have SPSS, and how am I going to go about? In Excel also you can use. So suppose, let me give only 1-2 example in Excel, then my talk will be over. So this is an Excel. Let me share. This is an Excel, and this is the same data I have given in 3 form. This is the no treatment nanoseria at 50 and nanoseria at those 300, and this is the mitotracheal signaling, which is the this is the one that is what you call with added and a no added nanoseria. So suppose if you want to do the unpurity test, suppose, or purity, how you are going to do in Excel, just click the data option in Excel here. You just click in data option, and you can see that there is a data analysis option is there in Excel. Just click there. In the data analysis option, then lots of tests are given, and which test you have to use? You have to use the for example, peer sample test. You just click here, peer sample. So you just click OK. Then Excel will require 2 group. Which one you have to define? Just click here, and this is the group 1. From here to here, I will just click and click enter. So that means it is a group 1. Another is a group 2. Group 2, you just click here and select here, and this is a group 2, and level, because you also included the level. So check the level here, p value, you have fixed the alpha error at 0.05, that is 5%, and hypothesize mean difference. Null hypothesis says that there should not be no difference. So you can put 0. That means hypothetically it will be 0. So you can click here, and you can get a what you call a beautiful this thing in the Excel itself. If you don't have SPSS, and you can see that the p-value 1 tail and a 2 tail is given, and p-value is 0.0035. In this way, you can just show the p-value. So lastly, I will just try to show how to make similarly in one-way ANOVA also you can do in here in what you call Excel. So and this is a one. Suppose if you want to do one-way ANOVA, then you just click here. The ANOVA data is there. See, one-way ANOVA, single factor means one-way ANOVA. You just click here, and in the range, you just click here. You select everything from here to here. And since you have also included the level, you select the level here, and then you just click the detail, and you can see the beautiful data which is given in the Excel output, and you can see the p-value. That is, p-value is 0.44, which is not statistical significance. So in this way, you can use the Excel to analyze your data also. So last, I will show how to draw a graph in Excel using error bar. The error bar is very important because Excel doesn't give you the error bar just like that if you go ahead. So for that, it's always better to calculate the mean and standard deviation first if you want to use a plus minus 1 standard deviation as your error bar. So how you are going to go ahead, just click equal to, and this is a short version. So you can see average. Average means the mean. You just click here, and what are the average which you want? I want average from here to here. So you just click here. So this is the 4126 is the average of this column 1. In the same way, if you drag like this, then you will get same formula will be applied subsequently, and you can get the average of the column 2, that is group 2. Again, another one is that I want to see what is the standard deviation. What is the short form of standard deviation? STPV. So ST, this one, STDEV. Just click here, double click, and it says what you want calculation of a standard deviation for which one. So you just go ahead and click here because I want standard deviation of this one. Just click here, and you can just drag it and see and just bring it to the your right side in order to calculate. So we have mean plus minus standard deviation, and how we are going to make a data. Just insert, select this value because you want to make a mean. Just go here. This is your what you call your data. So but it doesn't give you the error bar. You are going to go ahead, just click here if you want to see whether the data level, this is data level, it based upon a mean. You can do all sort of things in SPSS as well as in Excel also. If you want to say error bar, how we are going to add error bar, just click here, plus option is there, or you can click here also add element to the graph. So whatever you want, option is there. Let me just click here, and then you can see that the error bar is given. See, error bar is there. But error bar, you want to customize the error bar to plus minus 1 standard deviation. How you are going to do that? Just go to the click to the error bar here. Suppose where this small arrow is there, and in the small area arrow, just drop down and click more option. More option. So in the more option, you can see that customize the error bar option is there. You just click here because you want to customize the error bar. You want to use error bar plus minus standard deviation, 1 standard deviation. And where is the standard deviation? You have to tell the software to specify the software is asking for the location of your standard deviation, and location since I have put here, so you just have to click here, and then tell the what you call this thing that your this thing is here. That means this become your what अच्छा ये किसी ने click किया है यहाँ पे, खैर चलो. So I will again do it, same thing. This is your this thing. Sorry, let me check. So, but you want to put error bar not only in a positive direction, but in also negative direction also. In the same way, negative, same thing, you can tell the software where is the location of the standard deviation. You just put here, and you just put click or OK, then you can get the error bar beautifully in your graph. So this is the graph with inclusion of the error bar, error bar in plus minus direction. So in this way, you can use the Excel for your graphical representation, not only for analysis. So I hope this presentation was useful, and thank you very much. I have to rush like anything because time shortage is there. So in the future, we will hope to you please, all the participants are requested to request the organizing secretary to organize something in the biostatistics, also detailed workshop hands-on with all theoretical and practical aspect. Thank you very much.
Thank you, Dr. Barun. It was a speedy but elaborate talk on different type of biostatistical techniques. If you have any questions, please write them in the chat box, or we'll be unmuting you. You can ask the presenter.
Unmute. Okay. Please ask. You can ask about the earlier sessions also. If you have any doubt, we can clear them now.
If you have any more questions, you can ask us tomorrow as well. So tomorrow we'll be meeting again at sharp at 10, as we have been doing for the past 3 days. And tomorrow will be our concluding session. So I hope to see you all of you.
अनम्यूट नहीं की इसलिए कोई नहीं पूछता
Unmute.
कर दिया तो इसमें लिखा है ना अनम्यूट
We have unmuted from our side. Please check from your side also. नहीं अभी unmute नहीं है.
नहीं हुआ है अनम्यूट नहीं हुआ है
Please wait. We are correcting. What?
Yeah. Now we are unmuted.
Hello, sir. Thanks for the presentation.
Please
Can I audible?
Yes, we can hear you.
This is Mahesh from University of Hyderabad.
Yes, sir.
So one thing today I understood that you clearly mentioned that there is difference between p-value and confidence interval. So which is more prominent for the data analysis, as your opinion? I didn't get it.
Yeah, I got it.
Yeah.
Before going to there, you have to understand what is the definition of p-value. See, for suppose say a p-value of 0.03. Suppose if you do something and you get a p-value of 0.03. So what does 0.03 means? So for going into the interpretation of p-value, you have to do one assumption that is very important. Then you have to assume that null hypothesis is absolutely true. That means if you are doing some intervention and then you get a p-value of 0.03, that means how you are going to interpret that? You have to assume that there is no effect. Null hypothesis is absolutely true. Then whatever you are getting, because of which you are getting p-value of 0.03, it is just a random error. That means the p-value is basically tell you about the probability of the effect, which is because of the random error when you assume that the null hypothesis is true. That means p-value doesn't tell that your alternative hypothesis is true. Your alternative hypothesis means whatever you are doing intervention is right. So p-value doesn't give you about the strength of that. Suppose if you are getting p-value of 0.00001, and if you are getting one person is getting p-value of 0.03, it doesn't tell you about the effect size. How much effective is your intervention? It doesn't tell you anything about it. It just only tells that whatever you have set the criteria of 0.05, it is satisfying or not. That's all. That is the importance of p-value. So what all the important reputed journal, they are not going after p-value. What they require is you give the p-value up to 3 decimal point. Don't say whether it's significant or not, because many times what people do, people will just write NS, non-significant, or F significant. They don't mention the p-value. So always mention the p-value up to 3 decimal point, and always give confidence interval. Confidence interval is more important as compared to the p-value. And if you are doing some intervention, you have to give the effect size. Effect size is more important. Effect size means how much is the actual difference after giving your intervention. So that's why instead of too much focusing on p-value, it's time to move ahead from p-value and go into confidence interval and effect size.
Okay. I mean, it depends on sample size also. Always you are saying that. I mean,
absolutely. P-value depends upon sample size. See, the more if there is a small slight difference, suppose if there is a very slight difference and you are getting p-value of say 0.056. And you want to publish your data saying that the p-value should be less than 0.05, then how you are going to do that? You can just increase the sample size. Automatically your p-value will decrease. The p-value depends upon your sample size. So if you are doing a very small sample, even if the effect is so much difference, then your p-value may not be statistically significant. So p-value depends upon the sample. Similarly, power of study also depends upon the sample size.
Yes, sir. Thank you for the questions. And one more thing is, and you analyzed all this data in IBM SPSS, right?
Yeah.
So other than IBM SPSS, I think graphed prism also in general people use it.
You see, graphed is more user friendly. It's very easy.
So
it is very easy. Professional graph idea, though graphed prism is also very nice. You can go ahead with it. Even you can do in Excel.
Yeah. Can I perform all these activities in the same software?
Absolutely, you can do everything in the graphed prism. There are certain limitations in graphed prism. If you are going into a higher version, if you are doing like for example the analysis, principal component analysis, and all those stuff, then you have to do, step by step in Graphene Prism. Otherwise, Graphene Prism is extremely good, especially when you want your data, I mean, in graphical form, because the graph is very beautiful there.
Yeah. Thank you so much, sir. Yeah.
Okay. Thank you.
Any more questions? Is there any question in the chat box?
One thing, if you have any kind of questions related to PRP, also you can ask, because day before yesterday, many were asking about PRP.
Some were asking.
PRP, what is PRP, how to do, and all that stuff.
We can clear all those doubts from previous sessions also.
So I hope that we have satisfied the viewers. Now we will end the presentation here, and we'll meet again tomorrow. Thank you. Please end the
Okay, bye everyone.
Thank you, sir. Thank you, ma'am. Thank you, sir.